Hi, good morning, everyone.
Good morning, sir.
Good morning, sir.
Namaste. Good morning, sir. Good morning, Salahuddin, sir.
Good morning, sir.
Salahuddin, sir, your mic is off.
Very good morning, Dr. Ajit, Dr. Rakesh. Hope all you are doing fine.
Yes, we are doing fine, sir. How are you?
How was your? We are getting jealous that you had a very nice trip at Spain. After देख के जो है ना हमको भी we are also getting tempted now. We should visit. Okay.
Yeah,
Great,
the trip was amazing, sir. So I have visited 3 countries. One was Spain, and then I had one meeting, pre-conference meeting of ESC, European Society of Cardiology in Finland, Helsinki.
Very nice. Very nice.
Yeah, and then I had one day halt at Dubai also. So I went out and roamed around. Hello, ma'am. Namaste. How are you? How's everyone? Ma'am, your mic is off.
Good morning, sir. Good morning, everybody.
Good morning, Preeti ma'am.
Good morning, Preeti madam.
Good morning, everyone.
Okay, I think so, sir. If you allow, we should start, right?
Yes, sir. We'll start. Sir.
Yes, it is 10:15. Probably we have told 10:30.
Yeah, and there are a few candidates who are joining in YouTube also. So, okay, so we'll start at 10:20. So no worries. That's
Yes,
fine.
sir.
So by the time people, other people will also join. Yeah. So finally, this conference is happening. I'm very happy.
Yes, sir. Even we are very happy, sir.
Yes, sir. We are very happy.
Teamwork will always give you good results, sir.
Yes, definitely, sir. Let me see if Dr. Manoj is here.
Manoj sir has joined.
Yes, Manoj sir is joining in a minute. I just called him, so he's joining. Okay, so yes, I think Sarah joined. Yeah, Dr. Manoj is here. Hi, Manoj sir. Good morning.
Good morning, sir. Good morning.
Okay, can you hear, sir?
Yeah.
Okay, great. Perfect. Okay, so let's start, sir. What do you say?
Yes, we can start.
Yeah. So okay, I think we have a good number. We have crossed around 100 here, and in the live YouTube streaming also. So that would be fine now. Okay, great. So good morning, everyone. Again, I hope you all are doing well. So first of all, I would like to share the planning part of this conference before I go into this. We were planning this conference for so long. So long means almost I think 4 or 5 months. I was discussing with Professor Salahuddin, and we were planning something to do at Al-Ameen. And one day, sir told, like, let's do something different. Something different means very specific, which has not been done by me, because we are conducting some kind of general pharmacology or general pharmacy practice, or focus to the pharmacy practice things till now. So he told, like, okay, let's connect with our pharmacology department and do something in proteomic sciences. I mean, I was aware of the topic, but I was not aware in the this deep. So I was talking to Dr. Swati Mittal, madam. So she's the head of pharmacology at Al-Ameen College of Pharmacy. So we were discussing about what to do, and she told, okay, let's do something on principles and applications of proteomic sciences. I told how we are going to get the resource persons. because I was not aware. Frankly, I was not aware. And then we started brainstorming, and we searched everywhere, and we got actually many. We got many means we got from the industry. Okay, I request everyone to please put off your mics, all the delegates. So, we got a few from industry like Serum Institute of India, we got from IMTech, we got from, Bharat Biotech. And this conference was, first planned on the 5th May, if I'm not wrong, 5th May, yeah. And then due to some circumstances, because of Eid and because of exams, we postponed it to, 4th June. And when we looked into the 4th June, we did not get many things. Our resource persons were were not ready. We were again stuck with the things. And now then we thought, okay, finally let's do it on 9th. Still, guys, I'm so sorry. I'm sorry because our one of the resource persons from Serum Institute of India is not joining today. But I promise this session is due on me. Okay, this session is due on me very soon. I'll get him for you, and he'll be talking about the management and the genetic variations in the COVID variants. Very soon I'll do it for you. Okay, so today we are doing this conference with our 3 eminent speakers. Okay, 3 very eminent speakers who are very much specialized of this super specialized topic. We have with us Professor Saeed Ahmed. He's MPharm and PhD from Jamia Hamdard University, New Delhi. We have Professor Manoj Gadevar sir. He is MPharm and PhD from IIT Guwahati. And right now he's working as a professor at a School of Medical and Allied Sciences, KR Mangalam University, Gurugram, Haryana. And we have Dr. Preeti Sangwe with us. Madam is MSc in technology and PhD in technology, and she's working as associate professor at the School of Pharmacy and Technology Management and MIMS, Sirpur campus. So, today's conference is going to be, very big dhamal, because we have all the experts and they are going to talk on different topics on diabetes and, on, traditional medicine. And madam is talking about the history of proteomics and metabolomics, and she is going to cover the basics of the proteomics. So that is about the planning part of the conference. Now I would like to thank Professor Salahuddin sir, who is giving us this opportunity to conduct this conference at Al-Ameen College of Pharmacy. We could not conduct it physically due to due to the examinations and due to this corona protocols and multiple things. So definitely we are looking forward for some physical conferences as very soon at Al-Ameen, and that will be grand. We are planning something really big for all of you. Thank you, sir. And then I would like to come to our coordinators who are are doing really well. Professor Swati Mittal, she's only planning, I think, the entire thing. She's talking to our resource persons. She's getting them on board. She's planning the entire part. Thank you, madam. And we have Dr. A. Muthukumar sir. He is associate professor at Department of Pharmacology, Al-Ameen College of Pharmacy, Bengaluru. And we have our committee members, Mrs. Manjula G.S., Ms. Deepa R., and Mr. Dharmendra K. Sarma and Ms. Kavana Ji. They are assistant professors at Department of Pharmacology, Al-Ameen College of Pharmacy. So the entire team is working to deliver the good content to you guys. So I hope you all will enjoy today's conference, and I hope you are going to get a lot of things at the end. So that's all from my side. Thank you very much, everyone, all the audiences. I can see many renowned names here in my screen. So they are joining today to attend this conference. Thank you very much, everyone. And now I'll hand over this session to this mic to Professor Salahuddin sir to proceed further.
Very good morning. Thank you, Dr. Ajit, the CliMed team, as well as our Al-Ameen team, the entire coordination team under the leadership of Dr. Swati. Very good morning to all the registrants as well as to the attendees for showing so much of interest for this topic. I must say, when we were planning, as Dr. Ajit was telling, that we wanted to do something new, which we have an intention to bring something new to our attendees, because people are talking about something related to technology. We are talking some new topics, but we wanted to bring a topic which was not being touched or discussed. And that's how this we have been taught. When I said, Dr. Swati, that we have to do something from the Department of Pharmacology, believe me, the entire team got it into the action, and they had some 7 to 8 topics, and amongst that, this has been chosen, which has been endorsed by the CliMed team. And today, I must say that a lot of messages have flooded that this is one of the best topic, and we are attending, the leaders are attending, as well as even they told that the research scholars as well as the their entire PG students are attending. And I must say that I'm sure this is a topic which probably each one of us find it as a new, including me. I did see this as one of the newest topic, because something which we have heard, but we don't know that how this topic has been used as an application into our day-to-day life as well as into the health. And I feel that something discussing which we can evolve, because this pandemic has taught us that how our body immunities has to be increased, and how some new applications which has to be taken into the natural resources, and that can be taken as one of the way of treatment or as a protection for the human beings or for our health. And probably this was one of the topic, and today's speakers, they are all very renowned, they are experts in their own relative fields. And I must say that probably these 3 sessions of all the 3 speakers, Dr. Saeed Ahmad, an expert from Jamia Hamdard University, Dr. Manoj Gadewar from the Vishwakarma University, as you can see his profile from the VHU, he is as well as Dr. Preeti Sangave from NIMS University, Shirpur. These 3 experts, I'm sure, will clear the doubts, will give an information additional which will give or pave a way for exploring the additional research ideas into the people who are going to attend today. I'm sure the attendees will definitely, as I got to know the statistics, more than 600 people have already registered to this conference. That itself shows that how deep this information about this topic on applications of particularly the use of this proteomics is been in need. And as the topics has been very much been planned and it has been discussed, and today these speakers are going to talk about the DNA sequencing in the identification of the genetic risk, metabolomics in the identification and management of the pre-diabetes and diabetes, genetic mapping in type 1 diabetes, role of genome sequencing in identification of emerging SARS-CoV variants, and tools for metabolite profiling and early detection of diseases. And probably I think this is something a newer domain, though it may be related to the pharmacology. This will have a touch to every specialty, whether it is a pharmacology or a pharmacology, or even every healthcare worker wanted to know about deep into this topic. I'm sure this will have a big impact. Attendees do not leave in between, because there are certain aspects, doubts, whatever you have. We'll try to see that you get the fullest of the knowledge, and I'm sure the platform that is being provided through the CliMed by the Al-Ameen College of Pharmacy will definitely last long. I must say, Dr. Ajit, there are many university deans and the directors are contacting me. They are telling, sir, we wanted to conduct, you tell us how we can do it. And probably this has already been, a seeding has been done for this topic and for this activity.
Definitely,
I'm
sir.
sure many more top information activities will be provided through the CliMed. Al-Ameen will do, many other universities will join, and we will try to provide the best of the knowledge through this platform.
Yes,
I'm
sir.
very much thankful to Dr. Ajit Singh as well as to his team of CliMed solutions, for partnering with us in exploring and carrying out these activities. I'm very much grateful to Dr. Manoj, Dr. Preeti, as well as to Dr. Saeed for accepting our invitation and to be part of it. I know they are all very busy people, but in spite of that, they have given their time to speak on this relevant topic, which is going to create a lot of informative for this today's day, I'm sure. But I equally first thank to my entire team of the Al-Ameen College of Pharmacy, which has planned day and night in order to see that how best this can be carried out. Dr. Swati, Dr. Muthukumar, and all the other faculty members, Deepa, as well as other faculty members who has played a very vital role. I must thank all of them. Attendees will have a great day today. I'm sure this is going to create a lot of impact. Definitely, I'm sure the feedback that will be created today will give a way for conducting many more programs. Thank you very much. Have a great day. Thank you to all the
Thank
speakers.
you. Thank you,
Over
sir.
to you, Dr. Rajiv.
Yes, sir. Just nothing much, but just add on, I was talking to Dr. Preeti last evening. So she was, as I'm also not aware much into, this topic. So she was talking about that this can be used in the, outcome research. Proteomics can be used in the outcome research. So I really feel that, after today's conference, many of the students and researchers who are from the medical background, who are from the clinical pharmacy and pharmacy practice background, they will get some research ideas, and they will do something. They will pursue this topic for their further research in in the institutions. So I hope for the best, guys. Please learn well and stay till the end, because we have only 3 sessions. We are going to end by 1:30 or 2:30. Then you can even stay back with us for the poster presentations. We have poster presentations and our PowerPoint presentations, the abstract presentations from 2:30, 2:15 to 3:30. So, and one more, just a little information. So the feedback form will be provided at 1 o'clock at the end of 3rd session. So that would be like an attendance for the certificates. So whoever is attending the entire conference, so staying till 1 o'clock will be getting the certificates. So that's all from my side. Over to Professor Swati, and we can start with the sessions, ma'am.
Thank you so much, Dr. Ajit Singh. Thank you for the wonderful introduction to the seminar. And I think without wasting much time, we should start with the seminar. And as per the schedule, the first speaker for the day is Dr. Manoj. So our faculty, Ms. Vindhya, will be introducing the speaker, and then Dr. Manoj can continue with his seminar. Over to you, Vindhya.
Thank you, ma'am. So, good morning, everyone. I'm Vindhya NS, Assistant Professor, Department of Pharmacology, Al-Ameen College of Pharmacy, Bangalore. Welcome to the one-day virtual international conference on principles and applications of proteomic sciences on impact on metabolics. I would like to introduce our first speaker, Dr. Manoj Gadeva, Professor, School of Medical and Allied Sciences, K.R. Mangalam University, Gurgaon. Sir going to present on genomic screening of diabetes. Sir has completed M.Pharm in JSS College of Pharmacy, UP, PhD in IIT Guwahati, and he has 15 years of teaching experience in various institutions, have 29 research publications in various international and national journals, written 2 book chapters, and patented for design of anti-shivering scissors, and also applied or filed for another 2 patents, attended so many national and international conferences, and delivered lectures in many conferences and workshops. And sir has guided for around 6 PG students and ongoing 3, and 6 PhD students are ongoing. And sir also recognized at university level, and is serving as a member for academic council member and university research committee at K.R. Mangalam University, Gurgaon. Also a member of NAC Criterion 6, and also performed many administrative duties in PCI and AACP committees I would like to welcome Dr. Manoj Gadevar. So, please take over the session. So, thank you.
Thank you very much, ma'am, for giving introduction. So without wasting the time, just I will start the session. My voice is audible to all.
Yes, sir, it
Yes,
is.
sir. Yes, sir.
Yeah, yeah. So I will share the screen. So I'm going to deliver a lecture on genome screening of diabetes. So type 1 diabetes. So all we are aware about type 1 diabetes. So it is mainly caused by destruction of pancreatic beta cell, and it is a autoimmune disorder of beta cell. So in type 1 diabetes, insulin is no longer will be produced, which may leads to various signs as well as symptoms, or which may lead to hyperglycemia, ketoacidosis, and potentially,
Dr. Manoj, can
yeah,
you just make it to presentation mode?
sir, yeah, yeah, just a minute, sir.
Yes, sir. Full slides. Yeah, full screen. Yeah.
Yeah. Type 1 diabetes, it is mainly caused by
Sir,
dis-
it's not done. It's not done, I think.
it is
Yeah.
done. It is done from my side, sir.
Then I think, sir, you have to share that screen, because still it is coming, in the half screen.
It is not shared, sir.
Yeah. It is showing only one slide.
Yeah.
Now
Please
it is okay.
reshare, sir. Please reshare. Share again.
Just a minute, sir. I will share. Now it's okay, sir.
Yes, sir. Now it is okay.
Yeah. Sorry for the disruption. So type 1 diabetes. So it is an autoimmune disorder mainly caused due to destruction of pancreatic beta cell. So due to destruction of pancreatic beta cell, insulin will no longer will be produced, and due to non-availability of insulin, it may lead to hyperglycemia, ketoacidosis, and potentially death may occur if a patient will not be treated with insulin. So there are various treatment goals for achieving the normal blood glucose, that is by giving insulin, as well as if insulin will be given to the type 1 diabetes, there will be avoidance of long-term complications. So it is the second most common chronic disorder which is associated with childhood, and peak age at the onset it is around puberty. But type 1 diabetes can occur at any age, and incidence is increasing worldwide approximately 3% per year. If we are saying the incidence of type 1 diabetes, it is highest in Finland and lowest in Mexico. So there are various environmental risk factors. So seasonality at the diagnosis, migrants assume risk of host country, and there are various risk factors which are associated from case studies. So in that first, that is infant as well as childhood diet, virus exposure, hormones, stress, improved hygiene, and vitamin D. So concordance in the incident in the identical twin is always greater, that is monozygotic versus the dizygotic twin, and 15-fold increased the risk for first degree relative. When we are comparing the risk, it is almost 6% through the age of 30 years, and risk increases in presence of susceptibility of the genes. So most of the time what will happen, the type 1 diabetes, it is an autoimmune disorder, and antibodies will be produced which will destruct the pancreatic beta cell, that is functional beta cells, which are mainly responsible for the release of insulin. And mostly the occurrence of type 1 diabetes is always in the childhood ages, and that is mainly due to the susceptibility of certain genes. When we are talking about the MHC region of chromosome 6p21, that is major histocompatibility region of 6p21. So chromosome number 6 in that 21 is the 21. So what exactly will happen? This gene is mainly related with the immune system or the immune modulation. So predisposition to type 1 diabetes is better determined by haplotypes. So DRB1 and DQB1 haplotypes are more accurately determine the type 1 diabetes risk. So DRB1 and DQB1, these are the chromosomes as well as the haplotypes which will be responsible for testing or those genes or alteration in the genes may lead to the type 1. So testing for both the genes, that is DQA1 and DQB1, is slightly expensive. Means if we want to go for the alteration in the gene sequence of those mentioned DQA1 and DQB1, then it is a quite expensive method. So high risk type 1 haplotypes are DQA1 0501 and DQB1 0201. Another gene which is associated with the risk of type 1 diabetes, that is DQA1 0301 and DQB1 0302. So when we are talking about relative incidence in type 1 diabetes risk by the number of high risk haplotypes, so ethnicity, that is DQA1 and DQB1 we are compared. So here ethnicity is 2:1. When we are saying the relative risk of those genes in Caucasian, that is DQA1 is 16 while DQB1 is 4. African American, it is 45 and 7, while in Asian, it is 11:4. So when we are having this data, and we will able to know that it is having more prevalence in African American and less in Asians.
Wait. Hello?
Yeah. So absolute risk of type 1 diabetes to the age of 30 by number of high risk haplotypes. So when we are saying this number of high risk associated with the genes, that is DQA1 and DQB1 haplotypes. So ethnicity 2, 1 and 0. And when we are saying the prevalence of alteration in the gene, that is Caucasian, African American and Asian. So it is again more in African American as compared to the others. So amongst all these groups, Asian people or Asians are having less prevalence of type 1 diabetes, which is associated with high risk haplotype of DQA1 and DQB1. So risk of developing type 1 diabetes, it is 25% with two of them, that is DQA1 and DQB1. When there is an alteration or mutation or deletion in those genes, the maximum chances will be of development of type 1 diabetes, and 8.3% only associated with either DQA1 or DQB1. And if there is no alteration in DQA1 and DQB1, there is a prevalence of 1%, and there will be less chances, but other contributing factor may responsible for the development of type 1 diabetes. So these are the genome screen for type 1 diabetes. So IDD, that is insulin dependent diabetes, M1, M2, M3, and these are the specific chromosomes at which there will be either addition or deletion. But most of the time there will be a deletion which may result into certain immune response, or that may be associated with either the longer chain of chromosome or shorter chain of chromosome. And what exactly the things, that is Q is the shorter chain, P is the sorry Q is the longer chain, while P is the shorter chain. The first digit, that is 6, 11, 15, it indicates the number of chromosome, and the alteration which taken place, that is the
Manoj, sir, your voice is not coming now. I think.
Some technical issue.
Yes, yes, yes.
We'll be back.
Yeah.
yeah. So these are M1, M2, M3, M4, up to
Manoj,
yeah.
you have to make again presentation. Your
Sir, your presentation is gone. You have to share the presentation again.
Okay. Yeah, just a minute, sir. Sir, now it is visible?
Yes, sir. Yes, sir. It is.
So there are various genome which will be responsible for screening of type 1 diabetes. So in that M1 to M13, M14, M15, M17, M18, then PTPN22, VDR, interferon alpha, sorry, interferon gamma, and TGF beta 1, that is tumor growth factor beta 1. Then insulin dependent diabetes mellitus M2. So when we are talking about insulin gene, that is chromosome 11p15. So variable number of tandem repeats, that is class 1, class 2, and class 3. In class 1, the repeats were 26 to 63. In class 2, it is approximately 80 repeats. In class 3, 141 to 209 repeats, and relative increase in the risk, it is 2 times greater as compared to class 1 ally. Okay, so when we are having the class 2 and class 3, the repetitions are more, so the relative risk will be more. So class 1 is associated with lower mRNA in the thymus that may reduce tolerance to the insulin and its precursor. So as all we know that type 1 diabetes, it is an autoimmune disorder where the antibodies will be produced, and those antibodies will destruct the functional beta cells, and due to those degradation or destruction of pancreatic beta cell, a person may develop type 1 diabetes. So when we are talking about, or when we are comparing class 1, class 2, and class 3, the class 1 is associated with lower mRNA in the thymus, so that which may reduce the tolerance to the insulin and its precursor. So IDD M12, that is cytotoxic T lymphocyte associated 4, CTLA4, and chromosome 2q33. So it encodes a T cell receptor that plays a role in T cell apoptosis. So that may be THR 17 ALA and relative increase in the risk, that is 1.2 per 1.2, means what will happen if there is an alteration in the IDD-M12 genes. So in that case, what will happen? The risk will increase up to 1.2 fold, and dysfunction of CTLA4 is consistent with development of type 1 diabetes. So CTLA4, that is nothing but cytotoxic T lymphocyte associated 4. Then when we are talking about PTPN22, lymphocyte specific tyrosine phosphatase. So chromosome 1p13 encodes LPY, that is important in negative T cell activation and development. So this is mainly responsible for alteration and binding of LYP to cytoplasmic tyrosine kinase, which regulate T cell receptor signaling kinases. So as a pharmacologist, as most of the students or faculties, so all we know that there are various pathways, that is tyrosine kinase, tyrosine phosphate, as well as GP, means there are various signaling pathways. So in that case, what will happen? Cytoplasmic tyrosine kinase, which is mainly responsible for T cell receptor signaling kinases. So intervention trial for T1D, that is type 1 diabetes. So intervention, that is TZIGR, DIPP and TrialNet. So intervention is avoid CM, insulin and immunosuppressive agent. So target is FDR, GP genetic and FDR antibodies and genetics. So when we are having CM, that is cow milk, and insulin given which is by the nasal route, is mainly means insulin which is given by nasal route will be shown effective treatment, it shows effective action in the treatment of type 1, while first degree relative and GP is the general population. So natural history studies for type 1 diabetes, which is conducted in the general population, Daisy, Colorado, then Panda, Florida, and Teddy, US and Europe. So based on newborn genetic screening, that is the proper information consent, and parent are notified the result by giving on male, and general population at high risk, that is 5 to 8% recruited for follow-up, and more than 50% of children who will develop type 1 diabetes will not be eligible. So in that case, what is the thing? Most of the time, whatever the wards or whatever the children, they are screened for type 1 genome screening. So whatever the data we are getting or the data which is received, that will be kept confidential, and that when we are assess the risk, that is most of the time general population, they are at the higher risk for development of type 1 diabetes, and the prevalence is around 5 to 8%. So genetics and prevention of type 1 diabetes. So type 1 diabetes cannot be prevented because all we know that it is an autoimmune disorder. The prevalence or the severity may be reduced by giving some of the immunosuppressant drugs, because immunosuppressant drug, what they are mainly responsible for suppressing the T lymphocytes, and the immune response will be suppressed, and there will be no further degradation or destruction of beta cells. So education programs are need for parents for those children who are suffering from type 1 diabetes. Then we will move towards type 2 diabetes. So all we know that type 2 diabetes is also called as non-insulin dependent diabetes mellitus, and is a group of genetically heterogeneous metabolic disorder that causes glucose intolerance. So in type 2 diabetes, pancreatic beta cells are present, but their efficiency will be reduced. And those, due to reduced insulin insulin secretion, a person may develop insulin resistance. So approximately 90% of the individual with diabetes have type 2 diabetes. May be treated with diet, oral medication, physical activity, and the person who is suffering from type 2 diabetes may be asymptomatic for many years unless and until the glucose level will not be estimated and associated with long-term complications such as neuropathy, nephropathy, retinopathy, or microangiopathy. So all we know that various factors which are, various factors, so they are genetic factor, environmental effects, increase in the fatty acid level, and increased blood glucose level. So most of the time, a person who is suffering from type 1 diabetes, so will, again, there will be no glycemic control. Then second, environmental factor like exposure to various chemicals, mutagens. Then in hyperlipidemia, if we are talking about hyperlipidemia due to elevated level of TG as well as total cholesterol, a person may develop resistance to the insulin and there will be no proper insulin regulation. And finally, what will happen if there is decreased concentration of insulin in the blood, which may affect the blood glucose level, and there will be increase in the blood glucose level, which may result into insulin resistance and finally complications associated with diabetes. So there are various genotypes. So in primitive type, individuals who are metabolically thrifty were able to store high proportion of energy as a fat when food was plentiful, more likely to survive times. In recent years, a continuous supply of calorie-dense processed food and reduced physical activity. So what is nowadays, due to change in the lifestyle as well as calorie-dense processed food like fast food, this also changed the better glycemic control and due to reduced physical activity, because in type 1 diabetes burning of calorie and physical activity is necessary. So due to reduced physical activity, again there will be development of insulin resistance, and these changes are likely to explain rise in the type 2 diabetes. So type 2 diabetes, all we know that glucose, fasting blood glucose and random blood glucose. So fasting blood 70 to 100 and random blood glucose when it is more than 140 milligram per deciliter. When we are talking about the type 2 diabetes prevalence worldwide, in India it is highest, and when we are seeing it is lowest in the Poland, because most of the time India is a developing country where there will be changes in the lifestyle as well as due to day-to-day day-to-day lifestyle, there will be a maximum chances or there will be more chances of development of type 2 diabetes. So estimated number of adults with diabetes in developing country, as we know, India is a developing country. So the estimated number of people who are suffering with type 2 diabetes up to 2030, that the development of type 2 diabetes will be more in the age group that is 45 to 64, and it is lowest amongst 20 to 44. In developed country, if we are saying, most of the time 20 to 44, 45 to 64 and 65. So in developed country, the prevalence of type 2 diabetes is more after the age of 65, because in India most of the time what will happen, the type 2 diabetes remain undiagnosed, and the patient will develop severe hyperglycemia along with certain complication, and the diabetes will be detected at a very later stage, and at that time, to control or to have a better glycemic control, it requires more time. So most of the time in India, the affected population will be in the group of 45 to 64. So nowadays there will be increase in the prevalence of type 2 diabetes in children also. So those parents who are suffering from type 2, their children may have a chance to develop type 2 due to genetic predisposition. So environmental risk factor in type 2 diabetes, obesity. Obesity plays an important role in development of type 2 diabetes, and it is mainly due to development of insulin resistance, and around 80% of newly diagnosed cases of diabetes, that is mainly due to obesity. That is physical activity. Lack of physical activity also results into development of type 2 diabetes, and if we are going for better physical activities such as exercise, which will control the weight, improves glucose and lipid metabolism, which is inversely related to the BMI. So most of the time, impaired glucose tolerance in type 2 diabetes is approximately around 60%. And if we are modifying the lifestyle intervention, that may result into reduction in the glucose tolerance. Okay, and that is around approximately 60 to 70%. So when we are talking about genetics, when we are talking about genetics and type 2 diabetes, individual with a positive family history are about 2 to 6 times more likely to develop type 2 diabetes than those with negative family history. So there is a genetic predisposition, and those genes will be carried to the next generation, and the parents who are suffering from type 2 diabetes, their children is having more chances for development of type 2 diabetes, and for this risk is approximately 40%. And if parents are suffering from diabetes or they are having diabetes, the chances of development of type 2 diabetes in children's or in adults or in their childs is approximately 80% if one of the parent they are suffering from diabetes. So higher accordance for monozygotic twins versus the dizygotic twins. So it is mostly observed in monozygotic as compared to dizygotic. Has been difficult to find the genes for type 2 diabetes, late age at onset, polygenic inheritance, and multifactorial inheritance. So what are the genes candidates selected because they are in, what are the genes involved for the detection in type 2 diabetes? So these are because they are involved in pancreatic beta cell function, insulin action, energy intake, and lipid metabolism. So most of the time those genes are related with the pancreatic beta cell and those genes which are mainly responsible for lipid metabolism. Maybe they are associated with PPAR gamma, and the prevalence of type 2 diabetes, it is having a correlation with PPR gamma, that is peroxisome proliferative reactivator receptors, genome wide screen. So there is nothing to be assumed about the disease and etiology. So we are not aware about the exact etiological factors. So it becomes a difficult for finding a particular gene for type 2 diabetes and genome wide associated studies. So what are the various challenges in finding out those genes? So these are inadequate sample size, multiplex families, causes and control, difficult to define the phenotype, reduced penetrance, influence of environmental factor, and gene-gene interaction, variable gene at the onset, failure to replicate finding, and gene identified have small effects. Means at the end of study or at the end of genome screening study, a faulty genes will be identified, or those genes which are not having a particular impact in development of type 1 diabetes. Just a minute, sir. I think it has been disconnected.
Yes, sir. Your presentation is gone again.
Yeah, just a minute, sir. Maybe network issue, I think.
No problem. No problem.
Yeah. Now it is visible, sir.
Yes, sir. It is. It is visible.
So most of the time, what will happen at when a genome screening has been conducted, the genes, whatever the genes identified, they are having a less impact and small effect. So due to that reason also, that is also one of the challenge for finding a particular gene which is responsible for development of type 2 diabetes. So CAPN10 NIDDM1. So chromosome 2q37.3, it is mainly responsible for encoding and intracellular calcium dependent cytoplasmic protease that is ubiquitously expressed. So all we know that most of the time drugs, they are mainly responsible for opening of calcium channel, and these calcium channels are responsible for exocytosis. So the process of exocytosis means due to that reason insulin will be released, and this insulin will be responsible for regulation of blood glucose. So due to this protease, that is chromosome 2q37.3, it will be expressed and may modulate the activity of enzyme or apoptosis. So there is a programmed cell death, and due to that reason, there will be a chances for the NIDDM and likely to involve in insulin secretion and resistance. So just now I told you that uh, calcium means calcium ion channels are mainly responsible for regulating the contraction, and if there will be no proper regulation of calcium inside the cell, the process of exocytosis will be affected. And sometimes what will happen, there will be development of resistance, that is mainly due to insufficient release of insulin, and due to insufficient supply of insulin, there will be a chances of development of resistance. So stronger influence in Mexican American than other ethnic group, and it is responsible for approximately 40% of familiar clustering. Familiar clustering means those parents who are suffering from type 2 diabetes, there may be a chances of development of type 2 diabetes in their wards. So genetic variants associated with this, that is A43G, THR 50ALA, and PHE 200 THR, and estimated relative risk is approximately 2%. Then PPAR gamma. So peroxisome proliferator activated receptor gamma, that is chromosome 3p25. So transcription factor, they plays an important role in adipocyte differentiation and function. So alteration in the PPAR gamma may result into difficulty in differentiation, and adipocyte will not function properly. So it is associated with decreased insulin sensitivity. Means PPR gamma modulator, what they will do, they will reduce insulin sensitivity or insulin resistance, but due to alteration in PPR gamma, that may be results into insulin resistance. And target for hypoglycemic drugs such as thiazolidinediones, mainly the rosiglitazone as well as, thiazolidinedione. So genetic variant Pro12 ALA and Pro-E risk alive common, and relative risk is 1 to 3%, and it is most common. So PPR gamma alteration in chromosome chromosome 3p25, there will be a chances, means approximate 25% chances of development of type 2 diabetes. So ABCC8 and KCNJ11. So ATP binding cassette subfamily C member rate of chromosome, that is 11p15.1, and second one is potassium channel inward rectifying subfamily J member 11. So chromosome 11p15.1. So ABCC8 encodes the sulfonylurea receptors. Okay. So most of the time sulfonylurea, what they will do, they are mainly responsible for degranulation of pancreatic beta cells. Degranulation of pancreatic beta cells, and due to degranulation of pancreatic beta cell, release of insulin will take place. So it's coupled with KIR 6.2 subunit and part of ATP sensitive potassium channel, mainly involved in regulating insulin and glucagon release. So mutation affect channel activity and insulin secretion. So mutations associated with ABCC8 may result into the alteration in the activity of channel and insulin secretion, because most of the time what will happen, the beta cells are mainly responsible for release of insulin, but due to alteration or deletion in the gene sequence of ABCC8, it may affect release of insulin. Means insulin will be released, but to the less extent, which may develop into type 2 diabetes. And most of the time, ABCC8, this is the site for action of sulfonylurea, that is one of the oral hypoglycemic drugs. And genetic variants, that is serine 1369 alanine and glutamine 23 lysine respectively, and the relative risk, that is 2 to 4%, means the alteration in the gene sequence of ABCC8 and KCNJ11, the relative risk is around 2 to 4%. It is very less. Most of the time the alteration in the gene sequence is very occasional. Then TCF7L2, that is transcription factor 7 like 2, that is chromosome 10q25. So it is related to impaired insulin release of glucagon like peptide 1 and reduced beta cell mass or beta cell dysfunction. As type 2 diabetes is mainly due to reduced insulin secretion. So reduced insulin secretion may be due to the defect in defect in beta cell, or second thing, genetic predisposition, or third thing, reduced beta cell mass, and fourth one is, as already we seen with the last, that is alteration in the potassium channel and calcium channel. So stronger amongst all, means it is a stronger amongst lean versus obese type 2 diabetes. So most of the time 10% of individual are homozygous, and they have twofold increase in the risk relative to those with no copy of the variant. Responsive to sulfonylurea, not to the metformin, because these receptors, they are mainly responsive to sulfonylurea and not to the metformin, because most of the time metformin will be responsible for peripheral utilization of glucose, means metformin activate or metformin is mainly responsible for peripheral utilization of glucose. It is not having any action of functional beta cells. And genetic variant, that is RE790 1694 and other in LD, and estimated relative risk, that is approximately 1.4% due to deletion of some of the genes associated with TCF7L2. GWAS new loci identified, that is FTO, chromosome 16q12, fat mass and obese associated genes. So in previous slide, I told that most of the patient or most of the people who are suffering from obesity, the prevalence of development of type 2 diabetes is more, and it is associated with chromosome 16q12, and governs energy balance. Gene expression is regulated by feeding and fasting, and the estimated risk in the alteration of this chromosome 16q12 is 1.23%. Then another, that is HHEX IDE, chromosome 10q23 or 24 near to TCF7L2. So hemopiotically expressed in hemobox transcription factor in liver cell and insulin degrading enzyme IDE, that is insulin degrading enzyme. So due to alteration in this gene or in this gene, a specific enzyme will be produced, which is a proteolytic enzyme, which may degrade the action of insulin by hydrolysis. Means once the hydrolysis of insulin will takes, there will be terminating in the action of insulin, and that is mainly associated with IDE, that is insulin degrading enzyme, and having affinity for insulin and inhibit IDE mediated degradation of other substances, and having the relative risk of 1.14%. Then another gene, that is CDKAL1, chromosome 6p22, that is cyclin dependent kinase regulatory subunit associated with protein 1 and likely to plays a role in CDK5 inhibition and decreased insulin secretion. So ultimately what will happen, due to reduced insulin secretion, there will be development of type 2 diabetes, and the risk is around 1.12%. Another gene SLC30A8, chromosome 8q24, solute carrier family of 30 zinc transporter, and may have major autoantigen for type 1 diabetes. So the risk again it is 1.12%. Then IGF2B 2B P2, chromosome 3q28, insulin like growth factor 2, mRNA binding protein 2 regulates IGF2 translation, which may stimulate insulin action, and estimated relative risk is 1.17%. The next one is CDKN2A/B, chromosome 9p21, so cyclin dependent kinase inhibitor 2A, and it plays an important role in pancreatic development and islets proliferation. So due to absence or due to deletion of this gene may result into hampering in the development of pancreas. As we know that pancreas is having both exocrine and endocrine function, and the endocrine function of pancreas is mainly responsible for release of various hormones such as insulin, glucagon, pancreatic polypeptide, and this deletion of genes may also having a specific effect on islets proliferation, and if islets proliferation will not take place, there will be no release of insulin for insulin from pancreatic beta cell, and the relative risk for this gene deletion is around 1.2% for the development of type 2 diabetes. So type 2 genes are a drug target, that is PPR gamma, ABCC8 and KCNJ11. So drug used routinely in the treatment of type 2 diabetes and pharmacogenetic implications. So already I told that thiazolidinediones as well as sulfonylurea are the targets for those genes which will be responsible for giving a prominent effect, and response to oral agent may be related to one's genotype, And genetic testing may be responsible for identifying individual at a high risk for type 2 diabetes and guide for treatment regimen for type 2 diabetes. Means if we are going for screening of those genes or alteration or deletion in a specific chromosome, then we will able to identify a specific target, and once the target will be identified, it will be very easy for giving a specific treatment for type 2 diabetes. That is pharmacogenetic approach and individualization of therapy. So based on age, body weight, sex, as well as ethnicity, we will able to give a specific drug for the treatment of type 2 diabetes. Genetics and prevention, that is type 2 diabetes is preventable, and there will be various preventive measures. In that first one, maintaining a appropriate body weight, exercise, then new genes or screening will provide insight into the etiology, what are the exact cause of type 2 diabetes, where addition or deletion takes place, or what are the mutations, or where is the absence of specific genes in which chromosome will give us the exact idea into the cause of diabetes, that is genetic etiological cause of the diabetes. Then public health message and having the genetic testing. So challenges include predictive value of most of the test is low. How to communicate risk information, means even if we are identifying a specific gene, then it is unpredictive, or it means how we have to communicate with the patient and what is the risk associated with that particular gene for specific information. Then healthcare professional may not be able to interpret genetic test. Most of the time what will happen, even if you are going for detection of certain genes which may be responsible for development of type 2 diabetes, the health practitioner will not able to correlate those results with the development of disease. For that we need a specialized molecular biologist, molecular biologist. They may correlate the data and will be able to help to the health practitioner for the prescription of suitable drug for regulating the proper glycemic control. Then genetic testing may lead to distress. Some of the time what will happen, after getting the result, the person may think that maybe he is having a deficiency of certain genes, or sometime due to addition or deletion, that person may have the phobia that he may develop cancer in future. So there are certain distressing factors which will get through genetic testing. Then insurance and employment discrimination, confidentiality, and direct to consumer marketing for genetic testing. So these are some of the challenges which may be associated with the genetics. The last one, that is maturity onset diabetes of the young. So it accounts approximately sir, I'm having time 5 to 10 minutes. Hello.
Yes, sir.
Yeah. So it accounts for approximately 5% of type 2 diabetes, which may result into single gene defect. So autosomal dominance, inheritance, or multiple generation affected. So early age of onset, that is 25 years, characterized by absence of obesity, no ketosis, and no evidence of beta cell autoimmunity. So maturity onset, it is not due to obesity or due to beta cell autoimmunity. So hyperglycemia is often corrected by modifying the diet. So these are some of the genes which are associated with maturity onset diabetes: Y1, Y2, Y3, Y4, Y5, Y6. These are the specific genes: HNF4A, GCK, HNF1A, IPF1, HNF1, 1B, NEUROD1, and these are the proteins. Hepatocellular nuclear factor 4 alpha, glucokinase, hepatocyte nuclear factor 1 alpha, insulin promoting factor 1, hepatocyte nuclear factor 1 beta, and neurogenic differentiation factor 1. So mutations around 12 mutations, approximately 200. So when you are saying it is highest in the glucokinase, that is the loci 7p15 p13, and the gene which is responsible for mutations, that is GCK, and approximately 15%. So 65% of maturity onset diabetes is associated with MODY3 HNF1A gene of chromosome 12q24.2. Maturity onset diabetes in young, one is HNF4A. So transcription factor, it is expressed in liver, kidney, intestine, and pancreatic islets, and has been associated with type 2 diabetes. So it is mainly responsible for it controls gene involved in glucose, cholesterol, and fatty acid metabolism. So this transcription factor will control genes which are involved in glucose, cholesterol, and fatty acid. So due to mutation in those factor or in those transcription factor, there will be improper regulation of glucose, cholesterol, and fatty acid metabolism. Controlled transcription of HNF1A and several mutation splicing defect identified, which will account approximately 5% of all maturity onset diabetes in N cases. Then another that is glucokinase, only MODY gene, that is not a transcription factor and required for glucose metabolism along with insulin secretion. So it will be acting as a glucose sensor. Glucokinase is acting as a glucose sensor. So MODY2 is generally a mild form of diabetes. So approximately 200 types or 200 mutations have been identified with this gene, and VNTR nonsense and missense mutations, which will account for approximately 15% of the cases. So next one is hepatocyte nuclear factor 1 alpha 12q24.2. It regulates expression of insulin and other genes which are involved in glucose transport along with glucose metabolism. So if glucose will not be transported to RBC, liver, as well as muscles, there will be increase in blood glucose level. Due to improper metabolism of glucose also, a person may develop into diabetes and results in a severe insulin secretory defect, which may contribute to abnormal islet cell development. Another impact of mutation in the said chromosome, that is 12q24.2, may result into abnormal islet cell development and severe insulin defect. And more than 100 genetic variants have been identified which are associated with MODY3, and mutation in MODY3 are most common causes of maturity onset diabetes in the young, which accounts around 65% in the total cases. And next one is transcription factor that regulate expression of insulin, somatostatin, and other genes, mainly MODY4, that is insulin promoter factor 1, on 13q of 13q12.1. Mutations leads to decreased binding activity to the insulin promoter, and due to that reason, reduced activation of insulin gene is response to the glucose. If insulin gene will not be encoded, there will be no proper release of insulin, which may have improper glycemic control. And genetic variants include friendship, insertion and missense mutation. So most of the time, either there will be addition or complete deletion, and missense mutation, and accounts for very small portion of maturity onset diabetes in the end. So whatever the mutations which are associated with MODY4, that is insulin promoter factor 1, they are least amongst all other gene mutations. Then MODY5, that is hepatocyte nuclear factor 1 beta, required for the transcription and expression of variety of gene, and it is highly homologous to HNF1A, which recognize same binding site as HNF1A, which regulate gene expression, and individual have lower renal threshold to the glucose, and it rarely causes maturity onset diabetes mellitus. Then next one is MODY6, is a transcription factor involved in the differentiation of neurons, regulate insulin gene expression by binding to the critical motif on insulin promoter side. And few genetic variants, that is missense and nonsense mutation, and account for approximately 1% of all maturity onset diabetes in young cases. So summary, just I want to give you a summary for this maturity onset diabetes. So these genes are expressed in pancreas and play role in metabolism of glucose, regulation of insulin or other gene involved in glucose transport, and development of fetal pancreas. So most of the time, these genes are mainly associated with regulation of glucose metabolism. If faulty metabolism will be there, there will be no proper glycemic control. Some of the genes which are involved in transportation of glucose to kidney, liver, muscles, RBC, and due to affecting the glucose transport, there will be no proper check in glycemia. And the last one, that is development of fetal pancreas. If pancreas will not develop properly, there may be a chances, or there will be a chances, that is the proliferation of pancreatic beta cells will not be proper, which may hamper release of insulin in later stage. So MODY phenotype depends on MODY genotype, and knowing to genotype is important determinant for treatment. So these are the treatment strategies for MODY phenotypes, complications and treatment: D for diet, O for oral agent, and I for insulin. So most of the cases, so MODY6, it is severe and requires, that is a diet and oral agent should be given for a better glycemic control. While some of the cases we need insulin injection for having a better glycemic control. Thank you. So I would like to thank Dr. Ajit Singh, Dr. Salahuddin sir, Swati Mittal ma'am for inviting me and giving a talk on genetic variants in diabetes yeah.
Thank you very much, sir. And it was a really wonderful session, and it was very much explanatory. And you have covered all the genetic aspects of diabetes, type 1 diabetes, type 2 diabetes. It was really good. So I would invite now our audience, if they have any questions, they can open their mic and ask their questions to Dr. Manoj.
Hello, good morning, sir.
Yeah, good morning. Good morning.
Sir, Master Dr. Ashwini Mishra. I am Assistant Professor in Department of Pharmacy, Barkatullah University, Bhopal.
Okay.
Sir, first of all, thank you for such a nice presentation on different factors and the genes which regulate the diabetes, and like what are their prime role in it. So I wanted to know, is there any neuropharmacological approach which can be utilized by using these genes in pharmaceutical research in order to find suitable drug molecule, or probably suitable scaffold hopping technique which can be utilized for the treatment of these kind of disease?
Yeah. Very nice question. First time I want to say that, yeah, we can have a neuropharmacological approach, because there is a means when we are talking about the release of insulin, it is mainly regulated by neurosecretory cells. That is means when we are knowing that endocrine function of the pancreas, that is pancreas is one of the endocrine gland and mainly responsible for release of various hormones, and the hormones of this pancreas is regulated by pituitary gland, and pituitary is a master endocrine gland. There we can go for a pathway, or we can identify a pathway which is disturbing the means which is disturbing signaling of those signaling pathway. And if there is a disruption in the signaling pathway, means sometime what will happen, a person will suffer from type 1 diabetes without any autoimmunity or without any defect in the pancreas, that may be due to neurosignaling. So at that time you can have positive feedback or negative feedback mechanism that you can correlate, and and if there is a disturbance in the neurosecretory pathway of pituitary, at that time you can have the solution. And second thing means you are talking about pancreas, so we will have one approach, that is regenerative medicine. So scientists nowadays, what they did, they have developed artificial pancreas, and those artificial pancreas are transplanted in the patient who are suffering from diabetes, and found that better glycemic control. Even if we will able to grow the pancreatic beta cell artificially, and those beta cell can be transplanted in the deceased patient to have better glycemic control.
Okay, thank you very much, sir.
Yeah.
Is there any convolution neural network technique or deep learning technique could be utilized in this direction? Like suppose, like in the case of COVID-19,
Yeah.
we use the X-rays of different patients and we predict the condition of the severity in terms of developing the this machine learning model in like as a pharmacy candidate.
Yeah.
So is there anything which can be utilized in order to predict the severity of the pancreas of the patient, and like how long the technique how long it will take to utilize this in order to cure this particular thing, if there is any CNN applicability can be utilized in this section?
See, actually, what is the thing? Though a patient who is suffering from a COVID, you can have a sample, but unless and until we will not go for autopsy of those pancreatic beta cell, we will not able to know whether that cells are functional or non-functional, or is there any dearrangement. That means what we can do, we will able to test, or we will able to estimate insulin level in that particular patient. But in order to understand the molecular changes in that pancreatic beta cell, you have to go for histopathology. And unless and until we are not going for histopathology, we will not able to have an idea that whether that beta cells has been completely destructed, or is there any morphological change. And some other modern techniques also we have to use, like flow cytometric analysis. So flow cytometric analysis will give you some of the alteration, or what are the different gene levels, like that. So this is one of the approach. And second approach means you can go for molecular modeling. So in molecular modeling, you have to identify some of the targets, and you have to go for screening of those targets by using your lead molecule, whether that particular drug molecule is binding to those receptors or those ligands, and you have to go for study. So one of the software that is you can use, that is Schrodinger. So which will give you means you have to identify some of the targets, such as I told, PPAR gamma is there, then some that is alpha glucosidase is there, then some other enzymes will be there. So you have to identify those targets, and you have to carry out the binding study. And if it is showing the prominent result, then you have to go for the further in vivo or ex vivo pharmacological study.
Thank you very much.
Yeah.
Okay, any other questions? I think someone has raised the hand. Please speak, Dr. Sivasankar. Okay, if no more questions, we can go ahead. Thank you, Manoj sir, for the wonderful session. It was amazing. Thank you very much.
Yeah, thank you very much, sir, for giving me an opportunity to
Thank
have
you, sir.
a presentation. And
Thank you, Dr. Manoj. It was very enlightening. I'm sure this session will have many much activities to be carried out. It was very much interactive, informative. Thank you so much, Dr. Manoj.
yeah, thank you very much, sir. Yeah, thank you one and all for giving me an opportunity and presenting a lecture. Yeah, thank you.
Thank you. Over to Professor Swati. Ma'am, please go ahead with the next session.
Thank you so much. Manoj sir, it was a very informative session. You have elaborated on the different genes and their mutations which are responsible for insulin resistance. So we've learned a lot from you. And now we will have our next speaker, Dr. Preeti Sangwe from NMIMS University, Shirpur campus. And our faculty, Ms. Kavana, would be introducing the speaker and then handing over the stage to Mike to her. Ms. Kavana.
So good morning, everyone. I'm Ms. Kavana, Assistant Professor, Department of Pharmacology, Al-Ameen College of Pharmacy, Bangalore, here to introduce our next speaker, so Dr. Preeti Sangwe, Associate Professor, School of Pharmacy and Technology Management, NMIMS, who is going to present on systems biology of metabolic discovery in today's session. So Dr. Preeti Sangwe completed B.Pharm from
Kavana, you are on mute. Unmute it and please speak. Yeah.
Yes, he's on mute. Yeah.
Yeah, sorry. So Dr. Preeti Sangwe completed B.Pharm from College of Pharmacy, ND-MVP, Nasik in 1997, and MSc Technology from ICT University of Mumbai in 1999, and got PhD in Chemical Engineering from ICT Mumbai in 2004, and LLB from Jitendra Chauhan College of Law, Mumbai in 2016. And ma'am has 17 years of teaching experience in various institutions and has received grant of 61 lakhs from DBT Biocare for her research work entitled Novel Approaches for Improved Production of Antimicrobial Proteins and Their Applications, and also received grant of rupees 85,000 from NMIMS University for research work entitled Green Synthetic Approaches for Production of Nanoparticles and Their Evaluation. And ma'am has published around 18 research papers in national and international journals and presented around 25 papers in national and international conferences and has written 5 book chapters in Elsewhere and Springer Nature. And ma'am has got many awards for her achievements in research field and has guided around 5 PhD students and around 69 M.Pharma students and 3 MSc students from the various institutions. So ma'am, we welcome you and take over the session. Thank you, ma'am.
Thank you very much, Ms. Kavana, for the introduction. With your permission, I request, I take permission to start the session.
Please go ahead, ma'am.
Tell me if I'll share the screen.
It is visible, ma'am. You can make it full screen.
Yes.
Exactly.
Am I audible? And the screen.
Yes, yes, ma'am. You are audible, and make it full screen. That's all. Nothing.
Yes. So, welcome to this session. This talk, the theme that the organizers has selected is futuristic. Futuristic in the sense that something which is currently happening, and the theme is going to dictate the future of pharma and many related fields which are going to handle biological samples. Okay.
Priti, can you make it into presentation mode?
Yes. Can you make it full screen, ma'am?
It is full screen.
Ma'am, then you have to
stop the presentation and restart it.
Then you have to reshare, ma'am.
Stop share and then reshare.
Yes.
Now is it visible?
Yeah. Yes.
Now it's fine.
Yeah. Right. So we are going to talk about metabolomics. Okay. Now the title that I purposely put is Systems Biology of Metabolite Discovery. There are so many things that are in perspective that we have to discuss to understand the depth of what this subject has to offer. That is one. That is first is understand, and then after I understand, we have to apply it somewhere. We have to apply in research. We have to apply for some hypothesis. We have to get some outcome, some meaningful or some clinical outcome which is going to impact the population in general for their benefit. Okay. So what we're going to understand are certain tools which are used to decode the central dogma that we have or that we have known so far, and the puzzle, the jigsaw puzzle that is being posed by the biological systems across, whether you talk about prokaryotic systems or eukaryotic systems. All these systems, the biological systems, are going to offer some complexities. They are going to pose challenges to researchers in order for the researchers to help understand what these small molecules really mean, how we can use the current knowledge that whatever we have gathered from different fields to put into some meaningful perspective. So what do we expect out of this lecture? My lecture, what are we expecting? Some learning outcomes out of this subject. Now, something like what this subject or what this topic really means is what we are going to understand, or my lecture is being planned like that. Why do we use this in the first place? Right? What is there something new that is being offered by this theme or this particular topic? We are going to try and understand. Having known what and why, the next question that a student or a researcher should be asking should be, how do I go about it? Fine, I know the topic, I know the theme. Then if I want to really perform experiments in the lab, what is my starting step? How are we going to execute the the knowledge that we have gathered in a meaningful manner. And the last question that we plan to understand through this session is where I'm going to use it. So where I don't want to limit, we have like before, like where, as I mentioned, there are so many endless applications that one can think of. I would not like to limit the thinking of the students, young students who are there, that yes, this is the direction and this is the right way of doing it. That's not the research, or that's not the way research is carried out. Research is always carried out by asking questions why and why not. Research is always carried out for the benefit of the larger community and making meaning of whatever time and money we spend into those kind of activities. Before I proceed for the content, the heart of this presentation, I would like to take a quick poll among the audience, the participants, to confirm that how many participants are there who are, they can raise their hand, who are undergraduate students, if anybody are there. Undergraduate students, they can raise their hand, so that accordingly I can gauge the depth of the talk.
Yes, everyone. I think there are less than 50% undergraduate students. Otherwise,
Okay.
all
How
are
about
researchers.
postgraduate students, students doing MPharm? And okay, few of them. Good. Okay, so we have a mix of MPharm, PhD scholars, and maybe postdocs. I don't know. PhD scholars around. And definitely faculty, and we have some industry representative also, right? So if you understand the kind of audience that we have and the theme that we're going to talk about is a mix. Mix meaning it's a big spectrum, something who is a beginner of trying to understand what this topic is. You have a mid-level researcher, and then you have an accomplished researcher, maybe someone who is going to support or fund these kind of project, right? So having said this, now let us deep dive into this particular topic, right? So now metabolomics. Now, if somebody can just unmute and tell me, right, what is this metabolomics, right? What is omics? If they have heard of, if they heard of this, I mean, they have just heard the word and do not know anything about it, or they know something or they are practicing it. So if somebody can unmute, or somebody can maybe write in the chat box. They have heard of it. They know how do we go about it. Any of these answers to the questions which are put over here? If somebody can unmute quickly for a minute.
Ma'am, it is the study of metabolites.
Is obvious, right? It is obvious. But why? How do I go about? If you could elaborate furthermore, Mathura.
Furthermore means the metabolites is of course the breakdown of the breakdown material that we get. So it's just the study about the low molecular weight and the metabolites here, those things.
Right? Yes, it's bang on, meaning we are talking about metabolites, which are small molecular weight compounds found in biological matrix. Now the question arises, yes, it is a study, right? The study has to be with a purpose, and study has to be with some method, and study has to be justified, right? So here, if you talk about this topic and we have a diverse set of students, it is also going to, or it should be providing some insight of how do I go about and what kind of things are anticipated in this kind of advanced technologies that we are talking about, or super specialty, as the theme has been rightly put by the organizers, right? So if you look at this study, right, it is an integration of what? Yes, you said metabolites, but what is the source of metabolites? Something which is a biological matrix, which can be from a cell, which can be from different sample. It can be a biotechnology project, or it can be industry funded kind of activity. It can be human sample which has been taken out. okay, for the purpose of deriving or trying to identify or extract these metabolites, right? So if I want to extract these metabolites, I will have to take help of some instrument, some protocol, some methods, okay, by which I want to perform. We are expecting that yes, it will be performed in a correct manner without errors or without bias, and therefore it is going to be relied upon. This process will be relied upon instrumental methods of analysis, which means there will be representation by QC/QA kind of expertise. Further, fine, I get information like in the previous talk, as rightly in an elaborate and an extensive talk given by our first speaker, he told the theme is genomics. Fine, I can define genomics, but you can now understand the complexity that goes into getting that data and then processing that data, right? So this field also relies heavily on inputs from the bioinformatics topic or bioinformaticians, or their softwares and tools which are there. Software developers, that is the background, the entire analysis that has been done for these kind of data is heavily relied upon the programs which are written for executing these kind of data. Okay, and finally, fine, the data is processed, data is identified, and now we meaning researchers or clinicians or scientists have to make a meaningful interpretation of that. So this is the journey that we're going to discuss in the session. Now here there are two pictures. I'm deliberately keeping it simple, and then we're going to pick up to the complexity of what this particular subject has to offer. So we are aware of the word genome, right? Genome, something which is going to be the entire DNA sequence of any species on the earth. Okay, fine. We said that is, we went deep digging into this particular molecule. Molecule is DNA. And now that DNA is going to offer complexity. DNA comes out as only 4 alphabets as ATGC in a particular format or a sequence, but without processing, without, I mean, we know that there are 4 nucleotide bases, but what do I make use of this becomes the question that one should be asking. So here we have the same species, but now this species expresses itself in its life form in 2 different formats, which is nothing but now we can call this as proteomes. Now, these are the 2 omics which were there, which has been there quite around, I mean, quite some time. And then there are sciences, there are courses, there are industries who are running this particular science or this kind of scientific information, right? So you have caterpillar and butterfly, and now this is the transition of data also, which is happening. Now one might say that what difference does it make for a butterfly versus caterpillar? The difference is in proteomics. How does it execute, right? How does it decide that these proteins are required? And the branch that came out of genomics was proteomics, because genome was very abstract, right? It's dry information, but proteomics is something which is going to be fluid and dynamic information. And we are trying to correlate by such omic studies. Now, why butterfly? We should take our species, right? So one genome, again, human genome, and there are so many proteomes, right? Endless limit. So when you talk about a particular organ, you talk about a particular system, cardiovascular, like as the previous speaker mentioned about diabetes is only one component or one problem area in this entire proteome, right? Which is governed by, of course, the genome. And now you can understand the complexity that the challenge as well as the complexity that one has to face as a scientist or a researcher. Now, where are we, right? We were satisfied with DNA discovery. Then we realized that that is not sufficient. There's something more to DNA. And then the branch came out as genomics. That's very much known to everybody. Then we landed with something which is proteomics. Fine. We said, now we have to correlate because we have human genome. Now let us understand what does this genome really make. So we started extracting samples from different tissue, from different, under different protocols. And we started to analyze these protein sample and going back to the genome and starting to say that this is the pathway that is getting affected by the genetic background for the clinical condition that we are getting. The transit is nothing but there is another molecule which governs the transition of the data or the information or the central dogma, which is nothing but your mRNA. Now these facts are known, and we were happy that yes, this is the working horse. This is going to be the driving force for the next few decades, or this is how the research will be shaped. Now you realize that there are so much intricacies, right? Like simple molecule like carbohydrate dictate the protein in a very different manner in different species under different conditions. Then the challenge or a new branch came out, which was glycomics, right? Again, an omics kind of platform or technology. Then we realized that why proteins, right? Proteins is one of the phenotype. It's not the phenotype, right? So yes, people started to realize that we are, for our sake, for our benefit, we conveniently ignored another component which is also present in the sample. The same sample, the sample has mRNA, the sample has DNA, it has protein, but we, for our sake of convenience, we ignored metabolites, which are also in turn impacting all these molecules, the mRNA for that matter, or protein for that matter. So now the new perspective that the scientists have gathered is nothing but metabolomics, right? We are going to focus on that. Now one would say that today's theme is on future, that is metabolomics, or current and future. There's another theme that maybe the organizers can think of, which is now lipidomics, right? So it's an offshoot of metabolomics, that is, we are realizing that even lipid molecules, just like carbohydrate molecules, are impacting even these small molecules, small in the sense that these metabolites are also going to govern the functioning of the cell or the system under given condition, which can be environment, which can be drug, which can be some clinical condition. Now here we are talking about the newest kid on the block. Who is that, right? It is metabolites, as rightly pointed out by Madhura, right? So here we, as I mentioned, we got satisfied with genome. There's now a well set protocol of how to handle genome, extract genome, execute. You have next generation sequencing which are there, right? CRISPR is on the block, right? Which is now again going to shape the future in terms of how do I edit some errors in gene, right? Transcriptomics, RNA are there, but then given their nature, we have to correlate this information. We have to build the entire picture by putting all the pieces together. Proteins, as I mentioned, right, it's going to dictate how things look like in terms of phenotype. And along with protein, this is the new thing which is there, the youngest one under the omics, which is metabolites. Now, metabolites, what is fancy about these molecules? Why, out of all complexities, why do you want another complexity in the world, right? We all already have sufficient complex environment surrounding us, right? Now, the real challenge is, we did say these are macromolecules and you have these metabolites. These metabolites are under the influence of, right, diet for sure. Why diet? Because we are what we eat, and the food that we take will be now metabolized for different functionalities, okay, and in turn are going to impact the molecules which are going to maintain the cellular physiology, right? So diet, environment can be some external factors. It can be as simple like smoking, or medicine for that matter, okay, meaning somebody is taking a pill and now the levels will alter, and now can I really find out what kind of changes are happening in the cell. And the new thing which cannot be ignored, which is another hot cake in the research, is your microbiome. We have almost like trillions of cells which are in contact, right, in mutual or commensal kind of relationship per person, and therefore they in turn, in a very subtle manner, because we were, as I mentioned, we were conveniently ignoring it, like these facts, but now we have realized that there's something which is also there, which is around the corner, but we have not given sufficient time to explore it. So microbiome, human microbiome, is another factor which is in turn affecting metabolite. Now fine, we defined metabolomics as something which I want to monitor. Now if I say I want to monitor, we cannot ignore that who is dictating that particular level, and therefore we will try to understand that how do I gather the information and where do I use the information. So having known this, what is the main objective of, extracting metabolite, and I do what with that? So definitely what is going to be the future, it is going to impact the drug discovery. That is no doubt about it. Then there is going to be personalized treatment that we do have, like when human genome was to be discovered, we took almost like 10 years. And now you have advances in the technologies happening. And therefore, so can we categorize the population depending upon their genotype, depending upon their phenotype, and design a treatment strategy which will be more effective and minimum with the side effect. So can we have some better treatment outcomes is what the ideal goal that will be there, right, for any studies or the plans which are being carried out with use of all these omics technologies. And can I have better diagnosis? Can I stage a particular disease for like cancer, depending upon the level of metabolites? So, I mean, few slides which I want to talk about where are we as beginners or mid-level and where is the world going, right? So these are some couple of slides which are dedicated to what kind of applications are we talking about, which kind of field this science is touching. So there are terms which is called as targeted metabolomics and untargeted metabolomics, which we're going to see in a few slides later. But then if you see at this picture is talking about the rise in the publication. This is like some 10 years back. And this graph is always, I mean, towards the increasing side. So as I mentioned, proteomics was in the peak. Transcriptomics picked later after the proteomics. You have metabolomics, which is catching up. And that's why I labeled it as the newest kid on the block in terms of omics. And you see that number of publications per year across different scientific domains. What kind of domains are we talking about? Biotechnology, functional genomics, microbial interaction, pharmacology, nutrition, lifestyle, metabolic disorder, dysregulation, plant metabolism, huge. Any biological system, you will have some metabolite which can have some meaning to it. And health and disease is another important factor, which is rather important share that this study is being targeted. So like transcriptomics is assisting proteomics and genomics, right? Similarly, metabolomics is going to impact the previous omics that are already there in place, that is genomics and proteomics. Of course, with the help of advances in the computational methods. Now, where this is another slide purposely put because how does pharma industry look at this particular branch, right? So it's not, I mean, for us as in the sense researchers, industry where they are alert, right? They have funds, they have resources, they do think, they have a dedicated community who is going to think of what is going to be after 10 years or 15 years. So this kind of studies were there, rooted almost like previous 3 decades, right? But most of it was focused on toxicological and disease pathology, simple, right? Just extract the information and be satisfied with the information that we're getting. However, now if you look at in last couple of years or few years, you'll see that there's a sharp rise in the publication which are in this particular domain. And if you talk about the journal, kind of journal that it is getting published, it's Nature, Science, Scientific Reports, Cell, those kind of journals, prestigious journal that these kind of study are getting published. And if you look at the company side, these are all innovators. I mean, the companies who invest into R&D, right? And they would be extracting information, and if they would be rather redefining their pipeline, drug discovery pipeline, by using such technologies or technological advances. So what are the pharma industries into, and if you what kind of modifications they are doing by taking up these kind of projects in hand? Yes, now they are going to integrate the knowledge because we have vast amount of information in terms of genomics, proteomics, transcriptomics. The only thing, the link that is going to be there is going to be systems biology approach, or understanding the molecular basis of what is happening in the body. Understand pharmacological responses to the drugs, because that's where their workhorse is. That's where they're going to get their dividends on the research investment they're going to do. Plus, in a more ethical manner, they are going to try to bridge the gap between drug development. The main purpose is to give it to the population, right? So the gap between what the population or human body wants versus how robust the drug candidate should be is what the ultimate goal will be there by putting or integrating all these sciences together. So few examples how metabolomics has impacted before I go on how do I go about, right? So we are saying what, we are saying why, we are just trying to answer the second question, why it is there, right? So functional genomics, as rightly pointed out in the previous talk, is another data-driven kind of science, right? The logic is what do I do with the data? So the functional genomic branch did focus upon something like understanding the metabolic activities of genes, right? Annotate the functions, metabolic functions and their fate, or predicted for that matter. And now you have the holistic approach, which is now put into this particular science, which is called as metabolomic whole genome association studies, right? So you have functional relationship between several genetic variations within a population. So rather than one individual or one type of population, we are going to integrate that information through such kind of advances in the technology to do what? To discover novel metabolic pathways. Don't forget the logic is systems biology, which means we have to land up with knowing metabolite is one thing, but that metabolite belongs to which pathway is another thing. So trying to understand that. A few examples which students can find out in the literature. We can just mention one or two here, but then there is huge review and research papers which are there in this particular domain. Like for example, antimicrobial metabolite ecatonite in mammalian systems were detected by such kind of study, that is genome-wide association studies. Then identification of another enzyme which was in ribogenesis, right? Because they could now pinpoint, because the other metabolites were almost stable. There were only one or two variations or spikes which were there, which could be easily detected. And thus, these kind of technologies can help in further refining the information. Another example that I would like to cite is health and disease, that another tool or another field that in metabolomics is going to impact is your biomarker discovery. Now, biomarkers are something just like metabolites, like insulin for that matter is biomarker for diabetes. Or sugar levels is a biomarker of diabetes. So can I have some kind of validated targets, which can be a protein, which can be a metabolite also. And in such kind of domain, this science will definitely come into picture. In a landmark study, right, which was a turning point, or which has been a turning point, is a study which was carried out in NCI 60 cancer cell line. These are some standard cell line. Whenever there's an anti-cancer research being carried out, the metabolites, the drugs are being exposed to these cell lines. And metabolomic study was carried out over for 200 targeted metabolites in these different cell lines, meaning in vitro assays. And they discovered that there's a correlation, or in all these cell lines, there was one metabolite which was hinting towards something, which is glycine utilization, proposing glycine metabolism as a selective drug target, right? This is a very important and validated kind of information which came out of this study, indicating that if I can design my drugs against, because tumor cells are relying upon this metabolite, so even that kind of simple information can make a huge difference in terms of the way the drug discovery will be carried out in future. Another example, as I mentioned about microbiome, you have microbial interaction. I'll directly come to the example. For example, in this autism in mouse model, there was an oral treatment which was given with a microbiome as a human commensal bacteria, which is found in our body. That was given to the animal study. And it was found that because the population of the good bacteria increased in the animal model, there were changes which were seen in the behavior of that animal. So it could reduce the behavioral abnormalities and alter the levels up to almost like 34%. As compared to, effect of this organism on the metabolites was up to 34%, indicating that yes, there's a link between gut microbiome, host plasma metabolome, and even neurological condition of that individual. So totally, it's just one line, but you can imagine the impact that it can have on different kind of neurological disorders by just knowing these kind of study outcomes. This is for biotech kind of domain, just like pharma industry is looking at this science, even biotechnology, which are working with prokaryotic cell, fungal cells, it can be antibiotic production. Even for those kind of study or those kind of research protocol, we can use this particular field, right? Which will be correlating to different stress condition, which generally microorganisms are related to. I'm not going deep into it because most of you belong to pharma. So I'm going to skip this and we will now jump to how do I go about. The third question that we want to answer is how should I proceed, right? So as we came to know that it is integration of instrumental methods, right? We have biological system. We know that I can cultivate cells in the lab, right? In vitro cell lines. We have animal models, which is another source of metabolite, and definitely humans, right? A clinical study through which we can develop a proper hypothesis and clinically validate it, right? So most of the MPharm students, PhD scholars are aware of this workflow. What is this workflow? Quickly. If anybody can unmute. I hope they are listening. Which is
reply someone, guys.
Yes, yes. So somebody can unmute and answer. This is similar, like, I mean, most of you might be working on this or this part altogether, right? So left side indicates the favorite instrument, HPLC, which industry few decades, I mean, for few years, it was the workhorse, right? Hundreds of instruments in the pharma facility, and you would have samples being injected and data being withdrawn. Now there are integration, right? Integration of two instruments together. The next one is in the list is mass spectrophotometer. Now this instrument, the objective is simple, right? The liquid, I mean the HPLC is going to give me one aspect of my sample, which is going to be the peak, right? Which is going to give me some retention time and separation, right? Versus mass spectrophotometer of that same sample, it is going to give me another information, which is going to be mass of that particular substance. Fair enough. Two, single analyte and two outcomes of these two instruments, right? Integrated further. Now it becomes fascinating, right? Now my sample can be further correlated. I can have one sample being injected. I can have multiple samples being injected. These samples can be coming from different backgrounds. And now we try to improve the dynamics of this particular pair, right? The pair that is LCMS. Most of us, right, MPharm students these days are working with LCMS workflow, right? What is Metabolomics going to offer, right? The advanced workflow is adding more things to it, right? What kind of thing? Yes, it can be clinical data, so study cohort, different population, single like control and a particular treatment group, or they can have a longitudinal study over a period of time, data being gathered. Definitely the sample, even the type of sample, whether you talk about urine, you talk about blood, you talk about serum, about plasma, all these things are going to have different factors or different constituents, which in turn are going to dictate the outcome of the study. The measurements, the integrated instruments which are there, they are going to be driving this particular hypothesis or the plan that we want to do. Here, new things start, right? Here, it's not mass, right? It is mass for sure, but then we have to extract the information with respect to multiple metabolites, multiple small molecular weight compounds, and then make a meaning of it. So this meaning cannot be done without good knowledge or understanding of statistical analysis, right? Pharmacology guys or pharmacology faculty are very well versed with the biostatistical analysis which is being done. All data, whichever we talk about genomics, proteomics, any omics that we talk about is going to rely upon statistically validating the outcome. We shouldn't be biased. There should be minimum errors, and whatever information we're going to draw is going to have some meaning to it. We correlate this information with our hypothesis, we validate it, right? We interpret it. This is the challenge, or this is the objective for which this entire work will be carried out, and finally translate it if you're going to talk about data being derived out of human subjects, right? So putting into clinical practice. So metabolomic study provides a picture of every metabolite in an organism. Any sample that we're going to handle, the instrument is going to give me information about what kind of constituents are present in that sample, number one. Number two, it provides insight into metabolic response. Now here it is not the level. This level came out of an outcome. It can be diet, it can be food, it can be the lifespan, it can be age, it can be gender, right? There are so many factors, environmental factors, which are there, and because of which, as on date or as on time, I'm going to get that level of metabolite. So it is response to a biological situation or experimental manipulation. Whether I say control group or normal versus cancer, those kind of studies are going to hint towards some disturbances or some variation in the balance, the levels of the metabolite. That's the default information that I'm going to get. Further, as a researcher, we assume something. We say that whatever molecule I get, every metabolite will be measured, is what we anticipate out of that instrument. Whatever is there, it shouldn't be missed. If there are 50 or 100 or 200 or 1,000, my instrument should give me all those 50, 100, 200,000 out in quantifiable manner, and that the measurements will be biologically informative. This is the baseline. This is the rider on which we are happy that whatever information I'm going to get will be having some meaning to it. Okay, so as quoted in the previous examples, we saw that the metabolomic study has to be planned, okay, because we're going to handle so many things together. So many molecules together. We have to think of what do we want out of this particular study, whether I'm going to do it through an in vitro assay, I'm going to do it in an animal model, I'm going to do it in a clinical study. How much should be my sample size? When should I withdraw the sample? How should I store the sample? There are so many things which are to it, okay? And therefore, in order to make the study plan, the entire objective convenient, we can divide this science into two types, right? Targeted and untargeted. Now, targeted, as the name indicates, that we know that for sure that there are some specific levels which will get altered in this particular condition or under such environmental condition. So targeted, as the name indicates, we are focusing only on few known metabolites, right? So there are specific metabolites which can be primary, secondary, right, derived, getting modified. These kind of specific compounds are there. Now they will be quantified and compared. Obviously, you won't have a single population. You will have some control group, which is going to give us the changes what are going to have, which are there, and they are established with a reference. For example, let's take blood sugar level. It's a targeted metabolite, right? We know that there is some established in the sense there is some normal level and something which fluctuates, maybe creatinine or urea levels or blood sugar level. These are the one which we are going to monitor, and that's how we do it. And we monitor the selective transition. We compare with some reference standard, we compare with control, and that's how we get the information. But don't forget that the workflow will be starting from biological sample, the instrument, the data. In that complexity, that background is definitely there when you talk about even some known metabolites, but which can be like 50, 60, it can be around 100, but then those can be quantified. Untargeted is something which is a holistic approach, right? You analyze all detectable metabolites, something which my my solvent or the systems is capable to extract. Okay, it is going to run through LC. My mass is going to detect those, right? So I will get some known, which is very obvious, right? Because we know that what system we are going to handle, and therefore, and there'll be a few unknown, which is going to be more exploratory. That is some environmental factor which we didn't know. For example, in case of the cancer study that I told you, that was unknown, right? We know that there's a glycine thing, but the levels that are altering in the cancer was very informative, and that was striking, and therefore that led to the further information about how do the cancer cells use the metabolites, right? Definitely this is in discovery mode. We want outcomes, right? We want to, there is a chance, there is a trial and error kind of thing, and we do have to do it in a different population. Control definitely will apply also in these kind of study, that is, will have a huge, it can be population A, population B, or it can be control versus a treatment group kind of studies. So having said this, we now go to the basic understanding of what are the stages in this metabolomics, and then we will see where do I use it with some case studies. So planning definitely is needed, just like planning an event, right? We have to have what kind of agenda, what kind of audience, right? What should be the timeline? How should we, stage all the speakers? Those kind of things are there. So similarly, in my experiment, I have to understand why I have to do a thorough literature. What is the need of this particular experiment? Am I doing it as a Me Too kind of thing, or something which is going to be unexplored? I'm going to try that. So therefore, we have to have hypothesis, which kind of people, which kind of infrastructure, which kind of resources will be required has to be known, right? So that becomes a driving question in order to perform, because it is cost intensive, right? These instruments, even if I'm a humble researcher with no instruments in my campus or around, right, I will have to outsource it. Even I can have a good hypothesis, but I would need budget also to run this kind of show. The choice of whether I want to go the targeted way or the untargeted way definitely is governed by the hypothesis, the budget, the resources, the time. All these factors are important players when we want to work with this kind of, any research is actually driven by those factors. Concerning limitation in terms of throughput, the word throughput talks about how many samples I can handle per matrix, right? Do I need to have different time points in one animal? How many times I want to withdraw the sample, whether I do the study for 7 days or within a day, or I monitor over a month? How do I decide, right? So that becomes a throughput. Plus, if I have, say, if I'm withdrawing 10 samples in a day from one animal, and if I have, say, 30 animals in a study, so you can imagine the number of samples I want to handle, and I have to execute those in terms of metabolites. So don't forget that. So workflow is another factor that we're going to see. Sensitivity, metabolite degradation. We cannot ignore this, because just like mRNA, for that matter, mRNA has a very short lifespan. So proteomics is more reliable. Transcriptomics, if we land up with mRNA molecules, yes, it is going to be informative. So similarly, metabolite degradation, how stable is the metabolite? Is it dynamic, or it is changing or rapidly changing, right? Those kind of factors cannot be ignored, or we would be missing out on some important information. Intrinsic sample variability, right? Even the column, the instrument, the solvent, the reagent that I'm going to use, all these experimental setup is going to have an impact on what kind of metabolites I'm going to get out of my sample. Available amount, how much blood should I withdraw? How much volume should be there, right? How should I process it? Should I process it immediately, or should I keep it in -80 for a day or so? Because, for example, if I want to monitor ATP, now ATP has to be done in fresh samples. I cannot store it. It is not a tissue sample that I can store it at -80 and maybe over a week's time I can do it. It doesn't work that way. So those kind of metabolites I want to govern what kind of workflow, what kind of time plans you're going to have for the study. And of course, ethically, meaning you need consent if you're going to work with human population. Study design, like pharmacology people, clinicians are aware of what kind of study design, population, whether I want to do in a lab setup, I want to do it in animal setup, I want to do it in human subject. All these factors will be to be known and randomized in order to remove biases. Sample size, here the complexity goes on increasing, right? How much sample is a good sample? Is it going to represent the entire population, right? If I want to say clinically validate the metabolomic study, then I have to have a good sample size, right? I cannot work on 10 or 5 or 2 subjects or 5 subjects or 10 subjects and say that this is the study. Is not a good study design or a sample size, right? So sufficient amount of samples, available quantity, cost, right? Between biological groups, between control and those kind of things. If I'm going to work with an in vitro cell protocol and I want to execute a metabolomic study on cell lines, yes, the protocol for cell growth, right? Reagents which are used and the extraction protocol, all these factors has to be covered. How many, in what cell format I want to execute it. And if it's a clinical sample, then again, that is going to govern the size of this population that we're going to take it for study. Having decided, right, once you have sample in hand, then the care should be taken, right? Whether you're going to work with blood sample, plasma sample, whether you're going to work with urine sample. Urine doesn't pose much of a challenge, but blood metabolites, I mean, the metabolites in the blood definitely are going to pose a challenge, because from aqueous system, we want to bring it into the metabolites, the entire community out into the in vitro condition. And therefore, we have to understand what kind of challenges are there. So there is a sizable amount of time dedicated, something which is called a sample preparation, which can be, to give you an example, for example, citrate has to be avoided when studying central metabolism. Urine samples, they don't require some special condition or collection tube, which is the case in case of blood samples. If diet, because we are talking about metabolites, so whether it's a fasting kind of condition or a type of diet, even those kind of factors, because it's an external factor, is going to have an impact on the metabolite. After the sample has been withdrawn, you need to have, don't forget that volume, right? Volume and processing the entire batch that you're going to handle. Once you have, fine, you got the blood, the next thing that shouldn't be forgotten is that the metabolite should be stable till I inject into the instrument. So that transit, whatever is there, whether it's going to be immediate, whether it's going to be half an hour, half a day, those kind of factors have to be uniform for all the subjects or all the samples or biological matrix that I'm going to handle. That cannot be waived off, right? Once you get the sample, there's one important step which is needed, because you want to halt the process, right? So there's something which is called as quenching, right? Where we stop the enzymatic activity of these samples. If it's an in vitro kind of thing, we can add some organic solvent, we can do some temperature changes, we can do heating, thawing cycles. Acids or a combination of steps can work for stopping or quenching the reactions. Apart from that, if like trypsinization, I assume that many students might be knowing in vitro assays. So there's one step which is called as trypsinization in order to detach the cells from the glassware. Okay, so this is not recommended. This is a thumb rule which is followed in vitro assays, but in metabolomics, you have to just grab it and extract the content out of the cell culture glassware. The second important goal is extraction, right? You got the sample, now you have to extract it. So what kind of solvent next? And that solvent has to be compatible with the instrument, right? That pair, I mean, that logic has to be applied. So something like methanol water, methanol acetonitrile, those kind of things work. In presence of formic acid, those kind of simple combination can work in order to extract the molecular weight, low molecular weight substances or metabolites. We're not talking about protein here, exclusively small molecular weight substances. Now within that, we have a spectrum. That is another challenge that we cannot forget. I can have polar metabolites, I can have water soluble metabolites, I can have non-polar metabolites, and my sample, whatever extraction protocol is there, has to extract the entire type of constituents, the entire mixture which is there in the sample. For example, EDTA can effectively complex ions, ferrous ion, utilizing their catalytic activity enzymes which are going to use such ions. EDTA can be a complexing agent. You can add chloroform, you can add acid at low concentration. They can improve the stability of metabolites, and that's how generally scientists work in order to have a working time for processing a huge number of samples which are there. Now coming to, you got the sample, where do we have stored it. Now we come to what options are there, right? What kind of options are there? Yes, I can have, there's a image which is showing I can have sensitivity is my objective, separation is my objective, accuracy, right, precision, those kind of things are needed whenever we talk about instrumental method. So you do have combination techniques which have been adopted, like capillary electrophoresis and mass spectrophotometer, LCMS, GCMS, FTMS. So in terms of sensitivity, capillary electrophoresis is offering highest sensitivity versus NMR is another option with low sensitivity, but processing, of course, investment needed in those kind of instruments also cannot be ignored. High separation, all these chromatographic methods are going to offer good resolution and separation, which are needed for the further processing of that sample, that is ionization and fragmentation, right, which is done by the mass. So if those things cannot be, if we are even having a good chromatographic separation, I need to ensure that what kind of ionization I'm going to use it under mass spectra in order to get the spectra. And sample preparation will vary depending upon what instrumental method, what analytical method I'm going to use it for the analysis, right? So whether I'm going to work with ionic metabolites, I'm going to work with neutral, polar, non-polar, all these things have to be thought of. So I might have different columns applied in order to get different metabolites, and then we can execute for mass. Okay, now this is a, as we have less time, we want to cover, because it's a process, right? There are different stages into this. So we are just summarizing the steps. NMR, GC can be one option, or LCMS. GC generally requires, one of the major drawback, volatile is yes. Another drawback is it requires, if I want to do metabolite study, it requires derivatization, which can be challenging. And it can alter the concentration, which is adding a new step into my process. So can I have something which is straightforward, which works, that is for NMR, and LCMS works as a stable option, right, as a convenient option for doing metabolite study. So if I'm going to work with NMR as an option, there you can have metabolites which can be detected, can be up to 50 to 200 per sample, right? It can be amino acids, polar, non-polar, primary, secondary metabolites, sugars, volatile liquids, even large metabolites for that matter. LCMS, definitely the entire spectrum can be detected, but we have a wider range of metabolites getting detected through LCMS. So you, as students, you can find literature which covers all these three possibilities, huge amount of work which has been done. We are going to focus, one case study is on NMR and the other case study is on LCMS, which we're going to see in the end. Having said this, we have to minimize biases, right? And therefore, there is a mass spectrophotometry initiative which is there, which is just like ICH guideline is an initiative to normalize and harmonize whatever clinical studies are being carried out, the way we analyze the sample. Similarly, in order to have minimizing errors or bias, and we don't miss out on some important information or metabolites, there are some guidelines which have been written down, which indicate that how many times I'm going to run the sample, right? Multiples or replicates or triplicates are needed. I need to run blank, I need to run system suitability samples when I'm going to run with these kind of metabolite cases, rather samples, right? So QC, QA should be included in the analytical sequence at regular intervals, meaning if I am going to give 10 injections of my sample, the next one should be of my quality control. What is a quality control sample? Something which is known. I know the peak, I know the RT, I know the performance, how does it work under that particular column. So those factors are known, and therefore I can in turn gauge how my experiment is running, right? So within the sample, I can inject an internal standard, or during runs, I can have those QC samples which can be subject or which can be injected, right? So you have conditioning. There are just see the number of injections which are there. Conditioning injections, diluted QC series, meaning different concentrations we can check. I can have samples which are again randomized, or it can be pooled samples. Again, end it with another QC sample. So whatever stretch of injections you're going to do, you start with a QC sample and you end it with a QC sample and maybe blank or known references. So there is a guideline of what kind of molecular weight substances, how many QC samples or reference standards can be included. All these things are well written down. You got these samples, you're getting the information that you're looking at, right? So post analytical, the mass is going to give me different peaks, right? So generally low intensity signals they are studied, but depending upon what objective or what kind of levels of sensitivity you're expecting, we can have this kind of range which can be varied. So you can have these signals which are generated, plus the instrument. The instrument itself is going to offer some program, some software or data analysis program, which will help the researcher of how do I or what kind of interpretation I'm going to draw out of this particular spectra. Apart from that, given the impact the science, this metabolomic science is going to have, there are some government funded or government supported activities, and there are some public domain activities or information which is freely available for such scientific community to perform it online and then get the data analyzed. So there is something which is called as database, right? Mass MSMS database, which is, you can Google this information, NIST, which is supported by US, and you can have the standard peaks, right? What is expected for different, like almost like millions of compounds are there, and their standard spectra are already there in the database, right? What kind of peaks we are getting, what kind of fragmentation pattern we are getting, all these things is there as a ready reckoner. So what you can do, you can feed the information, you can screen the information, whatever spectra I'm getting, I can feed it to such kind of, or screen it, rather compare it with this database, and they will come out with some option that the molecule that you're looking at, the small molecular weight substance, is probably a particular metabolite. So that's the way the data interpretation, not interpretation, rather processing starts when we talk about MS. Apart from that, as you saw, the number of publications which is happening, which is this science has been picked up across, right? Across different domains, research domains. Plus, just like different information in genomics or proteomics, for that matter, genomics, right? It was put into public domain so that more science can be performed, or more research can be performed on such kind of information. Similarly, in metabolomics, you have freely available tools which can help you interpret the data, right? Or help you analyze the patterns or the trends which are there in the data, because I can have some picomolar concentration of a metabolite, I can have a femtomolar, but how do I correlate becomes a big question. And therefore, these software, as I told you, it's a integration of informatic tools also, software. There are, if you look at what kind of project the computer professional or they're doing these kind of project where they're writing programs, right? There's something which is called as Python and R, which becomes the background of writing these softwares for my data to be executed, right? So there is a language which is a program written in R, there's a program written in Python in order to convert the numbers into some meaningful information, right? So you have this data, which is, you just go to the website and feed in the information that you've got, and then it will help you to, it will put the data into some presentable or some different or very interesting manner, right? So that's, again, there are review articles which focuses on these kind of metabolomic data. So apart from that, you got the data. So when you're analyzing the data, it does some statistical analysis, something like these things are known to you, but where do I use it is the question that we're going to answer. So t-test ANOVA, which is like univariate analysis to see the comparison between the two kind of samples that we have got. So, and with a probability factor, the p-value is going to govern what is the requirement, or what kind of whether I can rely on this kind of information is what is evaluated.
Excuse me, ma'am.
Yes, sir.
Ma'am, can you please conclude?
Yes, no problem. Okay.
Yeah, thank you.
Couple of slides and then we are done, right? So this is another.
No problem. You take 5 minutes. Conclude in 5 minutes, ma'am.
Okay. Okay. Yes. So we have this is how the information is fed. These are some screenshots from the website which is being highlighted here. You can feed the column information, the kind of ionization strategy, instrument, right? Which kind of test you want to apply, and it will give a analyzed data. Data can be in the form of different charts, right? This is how the metabolites will be reflected, right? This is how it will be given out to the researcher. This is a heat map indicating the different levels of metabolites. If you see here on the right side, there are names of the metabolites, and the color indicates the intensity. So this is taken for diabetes versus healthy, heat map of control and diabetic patient in terms of their changes in the metabolite levels. Then there are some integrative studies which are being carried out. What is integration? Integration of multiple data points together and put it into a pathway, right? We are not going to stop at metabolite, but we have to correlate with which kind of signaling pathway or which kind of pathway is getting affected by such metabolites. So there are some plots which are there, and there's one, this kind of distance and the nodes which are there, which talk about the compound. They talk about the levels. The diameter of the circle indicates the impact. The distance indicates how far it is related, and you can have the pathway generated, which is going to take care of the system biology that is to be applied for your sample. So you have 3 stages, as I told you. The instrumentation part, you have what kind of ionization strategy is going to generate the fragments, and then statistical analysis and network analysis. It ends with that, and then you come back to your hypothesis. What is the thing that you want to correlate? So maybe one case study. There's one which is talking about antibiotic discovery. So you have the organisms being taken from different natural habitats. You have selected a strain, you grow it under different media condition, and you extract through different solvent. These solvents are now put to different instrumentation. As I told you, there's one example of NMR and the other one is of LCMS. So you can have NMR profiling. You can correlate, you can see the levels of metabolites getting altered, and that are correlated with new scaffolds which can be used for drug discovery is one thing which is really interesting, and industry is going to look at perspective, these drug discovery perspectives for metabolites, right? The other case study is indicating of Parkinson disease, how metabolite information is related with the Parkinson disorder, and whether to understand the treatment is really working with such kind of metabolite study. So here is a study indicating 3 population groups. They you can look at, it's almost like 460 plasma sample, right? Then they subjected to LCMS mode, right? They got different peaks, and they came out with, from each population, they came out with different number of metabolites per study, which were indicating towards something like indole acetic acid, phenyl acetylglutamine, etc. Lipid profile also was getting screened, and they came out with here that a metabolite panel of 4 biomarker candidates, which comprised of these molecules, were identified on the comprehensive sample analysis which was done, and it was indicating that if you monitor these metabolites for a treatment, it can be, the untargeted can be converted into a targeted study for validation. So we began with 4 questions, like what, why, how and where I'm leaving it for the audience to apply, right? We should not be limiting our information and knowledge. We have to apply it, and therefore we are concluding with the positive note that yes, there are some limitations, something like because there are so many samples being detected in a particular analysis, I need or we rely upon the software programs or the tools which are there. So these kind of challenges, though they are there, with different programs or software or in silico approaches, we can definitely overcome it. Not to forget, data interpretation is the key of this particular science, and hypothesis generation, that yes, it should come out with some valid knowledge, some biological impact is what is expected. If you look at what happens, the number of signals which are there in thousands of a typical LCMS data, contaminants can be there. There can be factors which are redundant, which is not required, which is not impacting anything. What we are looking at is something which is going to be unique. That's the challenge, and that's the way research committee is progressing. So future, whatever metabolites are there, there is an unknown relation, which as researchers we are going to try to find out. There is a need for, or the advances in the data interpretation, the way the graphs I told you, right, will facilitate hypothesis generation. And this has to be supported by statistical analysis, which validates whatever objective we had set out will be a true one, right? So there definitely, it is going to shape different industries, pharma for sure, and different fields are going to benefit out of it. So an urge to the research community that do explore this as an option, right? So thank you so much, participants and organizers. And if any questions, I'll be glad to answer.
Yes, thank you very much, madam. It was a wonderful presentation. Seriously, it was amazing, and you have covered almost everything from the history to the utilization and applications, and about the instruments, case studies. You have covered almost everything. Thank you very much. And now I'll ask audiences if they have any questions. Then we'll go to Professor Salahuddin sir for his comments. Any questions, audience?
Hello, good afternoon, sir.
Good afternoon.
Sir, actually, I do have a question. May I ask the question?
Yes, sure. Please go ahead.
Okay. Ma'am, thank you very much for this such a nice presentation on this particular topic. And it's really a hot cake in pharmaceutical research nowadays. Ma'am, I wanted to know that what are the in silico schematics which can be utilized for in silico research work in this direction, though like you have mentioned the websites or web servers where like we can get the data, we can curate it and we can utilize in our own purposes. But apart from it, there must be some kind of schematics which can be utilized to proceed in the direction of in silico metabolic work. So could you please explain it?
Yes, I mean, see, we are talking about a process, right? There are different stages of it, starting from your biological sample till final data interpretation. So there are statistical tools which will help you predict. As I told you, in the antibiotic kind of discovery, where we are talking about resistances being observed in the organism, right? That's the challenge. Now we are taking help of microbial community, which are capable of producing antibiotics in presence of maybe resistance gene, okay? But now we are not monitoring only antibiotic which that organism is producing, rather we are monitoring gene cluster. What is a gene cluster? Something which is, they are the genetic makeup which is going to dictate the production of these metabolites, right? So now these gene clusters are being correlated, that is, I use metabolomics data, I go back to their genomics, okay, and I come out with, yes, these are the genes which are getting silenced in general my when I'm growing the culture in the lab. Now the reverse way is, I know the gene clusters, I know that these metabolites can be synthesized by this antibiotic, I mean by this microorganism, and now we correlate or we try to extract these metabolites by inducing these genes and look at the the chemical scaffold that these novel metabolites are going to offer, right? Now, since it's a microorganism or it can be a fungi, they're going to give us some scaffold, and that becomes the basis for drug discovery. So then the in silico guys can take over. What is the link here? The link is I culture these cells, I go back to the gene clusters, right, unexpressed, and then I come back to my in silico, which is another traditional approach, or rather a well-versed approach which is there for drug discovery. So that's how it will be executed. This is one example, if I have satisfied your question.
Thank you very much, ma'am. And there is software Zestra. They have given this metabolomic study software-based model, and according to that, whatever drug molecule we are going to detect by pharmacophore mapping, we are utilizing this molecule for the generation of different metabolites, and on the basis of the toxicological functional group they are having, or probably the group which will going to induce some kind of disease like cancer or some other thing, it is going to give the prediction of it. So I mean to know.
We have to integrate it. We have to integrate the information. We have tools. They are there, right?
So could you please?
There, instruments are there, but then we have to correlate the information which is in a meaningful way is what is needed.
Yeah, well, there's only one last thing. Like, could you please share some softwares which are like unpaid, because like to go for Zestra and utilizing the software is like it's really
for metabolite study you're saying?
Yeah, definitely for the toxicological information of those metabolite, maybe in 2D or 3D form of the structural form, which could be utilized. Because like through PDPK modeling also we can go through the determination of these things, but nowadays softwares are really costly. You cannot use in your institutional research work in such a way. So if you have some idea about it, thank you.
Funded project, this works, but if you're a standalone researcher, in which case there are some, as I told you, packages are going to give me information on the metabolite information. I mean, as I told you, the heat map, for example, or the network mapping, which is there, which is done through free available packages. Okay.
Okay, like R package. Yeah.
Yes, R package, right?
Okay.
Within R package, R is not going to be, it's an umbrella, right? Umbrella kind of package. And within that, I can have a different objective for different kind of metabolites, right? So if you even just put R package, you will get a huge list. There are reviews which are there, excellent reviews, which covers on different tools and their objective. So even if we focus on that, we can end up using a freely available software that can work. If you want, I can share some links with the organizers which can be forwarded to you.
Sure, it will be great help, ma'am. Thank you very much, ma'am.
Okay, so now I'll go to Professor Salahuddin for his comments, and then we can go to the next session as it is lunchtime, 1 o'clock, and further questions we can take later.
Thank you, Ajit. Thank you so much, Dr. Preeti. I don't have words because the way you have covered and the information that you have shared on the topic, probably even it was an eye-opener, and for me also it's a new learning area, particularly with respect to
because we have a wide spectrum, audience. We have students and we have scholars, so we have to start from basic till case studies.
But the way you have covered, I must say that your teaching skills and your knowledge has really been made a very big impact for this presentation. And we are very glad and thank you so much, because this session was really motivating and inspiring and knowledgeable. And I hope many more sessions we need to have on this. We look forward to have your affiliation and association with Al-Ameen College.
Definitely.
CliMed. Thank you so much, Dr. Preeti, for sharing your wonderful knowledge. Thank you so much.
Thank you, madam. Thank you very much. Yes. Now I would go to Dr. Muthukumar. Sir, please go ahead with the next session, which will be taken by Professor Ahmed.
Yeah, thank you, doctor. My voice is audible?
Yes, you are.
Yeah. So good afternoon, everyone. So before I take this pleasure to introduce our eminent speaker, please allow me to express my gratitude regarding the conference, the backbone, the master brain of this conference, my respected principal, Dr. Salahuddin sir, and CliMed CEO founder, Dr. Ajit sir, and the backbone of the team. My HOD, Dr. Swati Mittal, and other team members like Manjula, Deepa, Kavana, Vidya, and Dharmendra for all their support and hard work are planning this successful conference. And come to the speaker, it's a great privilege and honor to express my gratitude to Dr. Syed Ahmed sir. Actually, I'm doing a research to identify the SAR profile. You're not able to get the full detail, but thing is, my beloved principal SARs have some detail. It's a shocking. I'm not able to guess it is possible. So like that. Sir is a professor in Department of Pharmacognosy in James Hamad University in New Delhi. And sir, I'm searching your profile or LinkedIn or somewhere. I got some Google scholar, Mr. Dr. Ajit sir sent to me. It's a really shocking, sir. More than 125 publication with index only. I'm not able to scroll. I got a pain actually to identify what activity you completed. It's very honor, sir. And citation is crossed more than 5,500. Even high index. It's very shocking. And sir shared some detail. You have guided, sir has guided more than 50 PG students and 15 PhD students. And sir has holding many projects and research grant like from DST, DPT, AICT, UGC, and Ayush. And also recently Shah got more than 10 crores grant from Ayush department. He also successfully completed the international conference so during the 2020. Sir also has a lifetime member in APTI, IPA, and SEF. So it's a great pleasure to introduce sir. You are a very good inspiration for a youngster like me. And stage is yours, sir. We are waiting for your inspiration speech. Thank you, my beloved principal, sir, and Dr. Ajit. Over to you, sir.
I think Dr. Saeed got disconnected due to network.
Yeah.
We'll wait for his rejoining.
Right.
Yes, he was here. Yes.
Okay, sir has joined. Yes, sir. Saeed, you can share. What do you?
So can you see my screen?
Yes. You are not visible. Your slides are not visible.
Sir, your slides are not visible. Your screen is visible.
Thank you very much.
One sec, I think. Sir, you can put your video off so your audio will be clear, sir. I think due to the low network, that is the problem happening.
Slides are visible, not yet.
Saeed bhai, you can go ahead. Voice little bit.
Okay, just a minute. Yeah, that's what I will do.
I think he is facing some network issues. He's joining again. Yes, now it is visible. Yeah. Yes, sir, it is full screen. Correct. So I think your mic is on mute, maybe. Yes, your mic is on mute side, sir. Presentation was visible. You can make it full screen, that's all.
Now slides are visible.
Slides are visible, sir. You just make it full screen, that's all.
Thank you very much. Thank you very much. And I'm very sorry for the trouble. In fact, we don't have light here in our department since last 15 to 20 minutes.
That's okay.
And yeah, that's why I have written you to put question answer session later. Okay. I switched from my desktop to laptop, and still it is having less battery. So I will move little faster. Okay. So, and
sure, sir, no worries. Sure, definitely.
Okay. So thank you very much once again for inviting me for this talk, and even Madam has presented good. Having a lot of medicinal plants, and going to talk on traditional medicinal plants.
Sir, your voice is again breaking. We are not able to hear it.
Working
Slaudin, sir, your mic is off.
So
on kariye. Yeah, you can speak. Mic on kariye.
Said, sir, your voice is not coming.
Then this is rank 1 Institute of Pharmacy.
Yes.
And little
आपका mic उधर ऑफ करिए.
सिस्टम
का mic ऑफ करिए.
I use traditional medicine major challenges and application of metabolomics in Ayush and in traditional medicine, as well as in the medicinal plants in 30 minutes. Okay.
Said sir, if any problem in
he is joining, power has come just now. He is joining. His department was having some electrical issues because of that network.
Yes.
Just now the power has been restored at the department. He will be rejoining.
Yes, sir.
Sir is planning from other device. He's joining from other device. Just wait for a few minutes. Has joined. Yes.
So, Ajit, can you see me?
Yeah, I am
yes,
audible right now. Yes, sir, you are audible. Perfect. Yes, yes.
Now things are perfect. It will move ahead. Okay. Because it was problem of that power shut down since morning, and that's why I was thinking that it may create some problem. Okay. Now
No problem.
it's okay. Okay.
Now it's fine, sir. Now it's fine. You are visible and audible perfectly.
So my slide is visible.
Yes, sir. It is coming. Yes.
Okay. 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 Thank you very much. Okay. For your patience. Who is this? Just take this.
Your screen has come.
So thank you very much once again for your patience. So I am screen is visible.
Yes, sir.
Yes, sir.
Okay. 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 So sometimes it happens in online. So I am working in metabolomic profiling of medicinal plants and traditional medicine. So we'll speak for about 45 minutes to 30 minutes, and in which I will tell you that how metabolomics can be applied to traditional medicine for its globalization and for global and other issues of traditional medicine, as well as of medicinal plants. We know that this is India is a mega diverse country and having a lot of medicinal plants and traditional heritage. And we are here in center of Delhi at the Jamia Hamdard, and then Jamia Hamdard is rank 1 institute of India in pharmacy. And in this presentation, I'm going to tell you about Ayush and some introduction about Ayush and traditional medicine, major challenges, and then metabolomics, introduction, and application in formulations and botanicals. So what we are doing in our laboratory, those things we'll discuss here. And you have been witness about development of global center, WHO global center and traditional medicine. It is at Gujarat, and that surely is going to help in promoting our Ayush system throughout the globe in different aspects and utilizing different traditional medicines for global population. Okay, and then if we talk about Ayush, Ministry of Ayush is composed of following: Ayurveda, Yoga, Naturopathy, Siddha, Suvarikpa, and Homeopathy. And about 15 lakh practitioners and traditional healers are associated with this system. Okay. And for the first time, it was developed as a department of ISM and H in 1995. Then Department of Ayush in 2003, and then Ministry of Ayush in Modi government in 2014. So if we talk about just one system, not going all in detail, so about Unani was, I'm talking Unani, working on traditional medicine. Now today talking on Unani, since we have just developed a center of excellence of Unani medicine here in our laboratory, and funded by the Ministry of Ayush. So Unani system is based on the principles of Hippocrates and Avicenna, and then we are here composed of 7 different principles of early faculties, and of also all of those theories, that is temperament and humors, mainly describe about different physique and medicatrix, that is study, and that's how they are treating very similar to Ayurvedic system of medicine. Not going in detail about principles, but in Unani pharmacopoeia of India, we have 2 types of pharmacopoeias. One is part 1 and second is part 2. Part 1 is comprising of mainly medicinal plants and with their monographs, whereas part 2 is comprised of formulations with their monographs. And then we have national formularies, and in those formularies we have hundreds of drugs and mentioned with their formulas. So we are talking about NFUM, that is National Formulary of Unani Medicine, that is having about 1200 formulations mentioned in them for different type of disorders. So if we talk about their quality control, so there are different type of formulations. Some are solid, some are liquid, and some are like powder and other type of formulation. So those formulations, as mentioned in NFUM, they have different parameters for their quality control, and like here you can see, so in them these are composed of different plant drugs, animals, and mineral drugs. So for quality control, we have for identification and physicochemical characterization, very basic parameters that we have for their quality control, but not for the quality control like complete quality control. That means complete identification of each and every metabolite. And that's what is the major challenge in traditional medicine and herbal formulations. And so metabolomic profiling, as I'm not going to define it right now, since Madam already told what is metabolomic profiling, will tell. So we using metabolomic profiling, we can do all those like standardization, quality control, pharmacokinetics, and then stability, and all those parameters. And even you know that these plants are composed of multiple components, and those even one component of that is one plant is composed of hundreds of constituents. So each and every constituent is going to play some role in the treatment of diseases. So they are working in multi-mechanistic way, and synergy-based approaches are there, and those all can be addressed well using metabolomics and network pharmacology. So see here just one example. If we are talking about quality control of modern medicine, so like a paracetamol tablet, when we are preparing a paracetamol tablet, say that it contains 500 milligram of paracetamol per tablet. So after preparation of tablet, what we do, we are checking it 100 milligram of paracetamol or not. Okay, so when we are analyzing them and finding out it contains 490 to 510 milligram, that is this plus minus 2% are variation, less than that, then it passes the quality control and reaches to the market. But what about ashwagandha or any of the traditional formulation what we have in the form of extract? Can we check the content of ashwagandha in the ashwagandha tablet after its preparation? So that's not possible. Okay, so but we can do it through metabolomic profiling. Why we cannot do? Because it is composed of hundreds of constituents. So quantitative estimation of hundreds of constituents is very tough. So since origin of metabolomics, so that we started working on this, and now we can say that there are different formulation to which we can analyze and identify each and every constituent present in them. Okay, so like a ashwagandha tablet, if it contains hundreds of constituents, we can identify each and every constituent and then quantify them. Okay, so that's how we can say that this is also quality controlled and similar to modern medicine, and that's what is the need of the day for globalization of traditional medicine and medicinal plants what we are using for treatment of different diseases. Okay, so this this is the example, like I told you, this metabolomics, so similar to genomics, transcriptomics, and proteomics. Like in proteomics also, that is that analysis of a complete set of proteins present at a particular time in a particular organism, and similar is metabolomics is analysis of complete set of metaboloids present in that organism at particular. So already you have been told that this is of two types, that is global or untargeted and targeted metabolomics. Using it in traditional medicine, like for dereplication, that is for identification of metaboloids present in extracts using high resolution mass spectrometry, and then metaboloid profiling, that how many metaboloids, like if you say about this oil, you are having many of the oils and doing for GCMS. So in that you are profiling that how much peaks of that oil, that means how much fatty acids and other metabolites are present. So that's what is profiling. So that in metabolomic profiling that we can do and identify each and every metabolite. Whenever we are doing metabolomic profiling, then we are getting huge data extract. When we analyze, we got a huge data, and for that huge data, it's useless until and unless we are going to analyze them with a statistical analysis. So we use some statistical analysis, and then it can be used for different purposes. How we can use it, we'll discuss it here. Okay, like here you can see this that Imblica officinal is collected from 8 different geographical regions, just in India. Okay, it is not having much differences, but in major metaboloids, but minor metaboloids are still having significant differences. And that's how when we analyze all those metaboloids, you can see that these are present in different quadrants. Okay, that means these are having different chemistry. So like that, we can in identification of differential metaboloids, as earlier speaker already told, that it can be used for identification of cancer or different disease condition. You know, in different disease condition, these metaboloids, some of the metaboloids are differing as compared to the normal metaboloids, and that's how we can, if we can identify those metaboloids known as differential metaboloids, and can be used for the identification of different diseases, or can be used for the identification of different chemotypes of different plants. So like that. So then we can use it for identification of molecular mechanism. How we will go ahead, discuss some experimental, demonstrate something which has been done in my laboratory in the next slides. So in revealing mechanism, and then in network pharmacology. So a lot of things that we can do through. Here, if we talk about traditional medicine, so in traditional medicine, you know that these our medicines are composed of different type of, like some of the medicines are composed of powder, some are juices, some are extracts, and maybe aqueous or hydroalcoholic extracts, some are distillates, some are unorganized parts, and some are minerals, some are animals. Okay, so the chemistry of all those metaboloids are different. Since the chemistry of all those, like extract, if we talk about aqueous extract, that mainly we are using in traditional medicine. So chemistry of aqueous extract is a little different as compared to that of hydroalcoholic extract. Chemistry of juices are different, and so chemistry of gums and resins and fixed oil and volatile oils are different. So depending on the chemistry of the raw material, that formulation or medicinal plant or the extract what we are using for the preparation of that formulation, so we select the type of technique like GCMS, LCMS, HPLC, HPTLC, and other techniques for performing metabolomic analysis. Okay, so in our laboratory mainly we are using LCMS and GCMS as well as HPTLC and HPLC methods for, so similar to that. So if we see other things also, I told you that these traditional formulations are composed of like solid, liquid, semi-solid, and gaseous dosage form. If we talk about gaseous dosage form, so those mainly composed of volatile principles. Okay, so the chemistry of those volatile principles are different to this form, like sugar-based formulation. So some powders, extracts are mixed with the sugar-based formulation. So it is composed of a lot of sugar, but little of those constituents. So before analyzing that, sugar must be removed. So sample preparation is a critical step in analysis of those that traditional formulation. Similarly, liquid-based do this form. There are some of them are composed of aromatic waters, that is arqiyat. Some of them are sharbat. So if it is a sharbat, that means it is having water soluble principles. So we are not going to polar metaboloids, but we are supposed to analyze phenolics, flavonoids, and glycosides, and other polar metaboloids, and if it is water-based formulation. And similarly, if it is aromatic water, so aromatic waters are usually having aromatic component, and we are supposed to analyze sesquiterpene. So on the basis of those things, we select different techniques for the analysis. We are talking about Asava and Arista. So Asava and Arista is very dilute preparation. So even if we are not going to concentrate them, we could not the desired metaboloids. And similarly, so since they have different chemistry and different bases, so sample preparation and selection of technique is very important. A lot of different techniques that we are using. Flo already told by madam. So here, this is one. If we talk about targeted metabolomics, so here some students of my laboratory had developed some methods for analysis of hydroalcoholic extracts of and extracts of Pistacia integrima and Quercus infectorius. And the purpose of study was that since Pistacia integrima is used in traditional medicine since a long time, but it's too costly, okay, and not available. So there are different traditional literatures who are reporting that you can use Quercus infectoria, terminalia, chivula in place of Pistacia integrima. So whether we can use it in place of as a substitute of Pistacia integrima or not, so far doing those things, we should go for the chemistry and then biological activity as well. So in chemistry, we should go for first targeted metabolomics and then untargeted metabolomics and find out how much percentage of aqueous extract of these plants are similar, and so that we can and what are the exact metaboloids active present in them. So whether the things which are written that you can use in place of this thing is right. So like that. So we are creating some scientific proof of those literatures which are mentioning these claims. So in fact, validating scientifically the claims reported in traditional medicine. So that's what we are doing in that one exercise that we did, that analyze a different metabolites present in them, like coffee. So many of them, 16 metabolites get together, okay, present in all those three, and but a few 8 of them were analyzed and the rest were not present. So like this we did, and how much percentage of them is present we analyze. So then we thought to, because this formulation like aromatic medicinal medicines, how we just prepare traditional medicine, and those distillates, those are composed of sesquiterpenes, and those sesquiterpenes, in fact, that how those best can be GCMS or GCMS. So not audible right now?
Sir, audible, but your
audible.
Yes, sir, you are audible, but your presentation is gone. Please share again. Yeah.
So your presentation is not visible and your voice is also not audible right now. Yes, it is visible, sir, but your, I think, mic is off.
So mic is on, I think, right now. I am audible right now?
Yes, sir. Okay. Yes, sir, you are.
So we started working on aromatic waters, and in the aromatic waters, what we did GC-MSMS analysis and identified so we identified the major metaboloids, and after identification of major metaboloids, what we did the pattern recognition analysis. And in pattern recognition analysis, what we do, we give it to the rats and take plasma after some time and in different time intervals and analyze the metaboloids, okay, and find out how many of them are bioavailable, okay. So a lot of work we did with all those formulations, did their pattern recognition, and then find out the metaboloids bioavailable. Out of them, the major bioavailable metaboloids we identified in each of those formulations, and did their pharmacokinetic analysis. And then stability was also the problem.
So again,
yes,
voice is some problem.
Okay, just a minute. Then we do the metabolomic profiling out of, like, just one example is Arke-McCoy and Arke-Kasni. So in Arke-McCoy, we found that 56 metaboloids were present, and if we analyze it, the GC-MSMS, and further, if we find out how many metaboloids are bioavailable, so it is about 38 metaboloids were bioavailable. Out of them, we find out the major 3-4 metaboloids and did their other activities, how we will tell you in the next slides, okay. Then we did some metabolomic profiling of Burhavia diffusa. So since it has been so used in different disorders, so what we did, it is used in nephroprotection as well, okay. So different cardifolia, so that these two plants were most active. Then we identified the metaboloids present in their extracts, and those were used in the form of aqueous decoction. So we prepared the decoction and prepared their non-polar and polar, fractionated their metaboloids, and those metaboloids we identified different metabolomic profiling. And then after identification of those metaboloids, we find out that how these are going to interact in Swiss ADME software, and showing how many of them are going to be toxic or safe, and how they may behave in vivo conditions. And then we found that there are different genes which are involved in the pathophysiology of kidney diseases. Constituent out of them, 10 of these metaboloids, whatever we identified, those are going to interact with the pathophysiology of kidney diseases, and these can be useful for its protective, kidney protective effect. So similarly, we did it for Tinospora cardifolia and Borrelia diffusa mixture. So what we did, since it was only just decoction was used, first we thought that we could find out the antioxidant compound by bioautography, and then after bioautographic identification of those metaboloids using LC-MS, we identified the gene ontology and find out how many genes are further interacting with the pathophysiology of kidney diseases, and how these metaboloids, whatever we identified through bioautographic analysis, how they are going to interact with those genes, and that's how they are going to have So we did that. Since the decoction was used, we prepared the aqueous extract, did toxicity studies and find out it is safe, and prepared the fraction, and then those fractions also studied for antidiabetic activity in vitro and in vivo models, find out the butanol fraction was most active. Then we did the LC-HRMS studies, metabolomic profiling of that butanol fraction, and identified all those metaboloids present in that fraction. Further, we did in vivo pattern recognition and find out how many metaboloids are bioavailable. In those studies, we found that some of the metaboloids are present in the plasma as a ghost molecule. Those were neither present in the butanol fraction and nor in this. So from where it came, so like this, we did a study and found these butane, isobutane, quercetin, and other metaboloids are having better activity. And then these are the total summary of metaboloids, 158. So in 1 hour, 2 hour, and like this type of metaboloids, and how like 27 metaboloids were present in that fraction, and then 15 were bioavailable, and then 6 more were bioavailable from gut microbiota, hepatocytes, so like total 21 bioavailable. Out of those 21, if we do in silico studies, we found that these these three metaboloids who are having most major compounds and having most of the activity. We did the pharmacokinetics and postulated mechanism, published and got patented. So like that, we do this type of metabolomic profiling studies in our laboratory. Similarly, we did for some plants and formulation. A lot of studies like we do in Pancharista, Triphala. So we did do HPTL, start from HPTLC, do HPLC, and then LCMS and GCMS. So in some Triphala formulations, we quantified about 14 metaboloids like targeted metabolomics. Similarly, we analyzed different cardioprotective plants like Emomum, Subulatum, Coriandrum, Sativum. So all those metaboloids we identify, like even one formulation like Pattarfudi is used in traditional medicine. And for urolithiasis, we developed, since it was used in the form of powder, we developed in the form of tablets, since the dose was about 10 grams, reduce it to 800 to 900 milligrams. Did the chemo equivalency by metabolomic profiling and did the clinical trials and other studied. Similarly, reduced the dose of Carica papaya and that is used in dengue fever related thrombocytopenia. Thank you very much, all of you. In fact, this trouble was because of some power problem in my laboratory, and even this internet connection. But I hope I would have delivered something and may be useful for you. So we have just one conference very soon, that is in July. And that is on multi-therapeutic therapeutics in Unani Ayurvedic medicine, and in association with ISTM. I invite you all to join that.
Yes. Thank you very much, sir. It was great presentation. Of course, there were some technical glitches, but it was amazing presentation. It was very nice. And now I would like to invite our audience to ask the questions before going to Professor Sultan.
Professor Anna Chandran, you wanted to ask?
Very good afternoon, Doctor. Can you hear me?
Yes,
sir. Yes, sir.
Very good afternoon to all the resource persons of today's webinar. It was given excellent by all the presenters for this today's program. And lastly, our Dr. Syed Ahmed, he has given about the metabolites of medicinal plants, natural origins. Presently, I just want to mention, and it's not a question, I just want because of cancer is increasing in a multifold all over the world. Two formulations like the turmeric and grapeseed extract, the metabolites of these compounds, turmeric, for example, curcumin, it has been well accepted. Seed and grapeseed extracts are all well accepted. It is in practice because now we are doing a lot of innovative research works. And what is the ultimate benefit to the common person who are suffering from the disease? So, Dr. We are knowledge for the benefit of the society, and now we have come out in our research institute at Bangalore. Cancer hospitals to stop the further progress of cancer cells, and similarly flaxseed extract and grapeseed extracts. So these are a benefit of our society to particular medicines for diabetes and for other hypertensions from natural sources. The metabolites mainly at alkaloids, glycosides, flavonoids, and Dr. Syed, you was telling about the quercetin, which is from red onion. It has got a very good antioxidant property. So these are the some of the extra points I just program. So with this kind of information, I hand over the floor to Dr. Ajit Singh.
Thank you, sir.
Thank you, Doctor.
Thank you, sir. Any other comments or suggestions or questions? If not, then over to Salahuddin, sir.
Yeah, thank you so much, Dr. Saeed. It's always great to listen you. Probably, I think after looking into technological difficulties, what we have faced today, having offline is another one which we should always look forward. We should always have both blended mode. We will see that next time Dr. Saeed participate along with us in an event that we conduct along with CliMed in our campus at Al-Ameen College of Pharmacy. Dr. Said's achievement is numerous. It cannot be described in 1 or 2 minutes the way he is carrying out the research at Jamia Hamdard, and the way he has got collaborations with the research projects. There are only few in numbers in India whom we can count that the government or the other sponsoring bodies are sanctioning projects to the tune of 10 CR. I think hardly you can find 1 or 2 people who are getting such a big project of 10 CR, and we can see Dr. Saeed one amongst them, and his research work is excellent. We have seen for his publications, his patents that he has carried out, and I have seen his leadership because I was part of the SFE, Society for Ethnopharmacology of 2020 in the 8th CIFEC, where he was the organizing secretary. And the last image which he has displayed, he was the hero to which he has made it a successful event, and we could see the traditional healers making part of it. The international attendees were part of it. So he is again inviting to all the people, to all the attendees to be part of his STEM conclave, which he is going to do it in July 2022. I'm sure Dr. Said's lectures was the insight to which he has delivered. I'm sure once again we look forward, Dr. Said, for many more such events to collaborate and carry out a larger activity at Al-Ameen along with CliMed as well as with Jamia Hamdard. Thank you for with all the difficulties. In spite of that, you did not leave the session and you still continued with the enthusiasm. Salute to your passion, salute to your knowledge, and thank you so much for being with us, Dr. Said. Over to you, Dr. Ajit.
Yes, thank you very much, Said sir. It was definitely, there were some glitches, but the presentation and your research work is amazing. You are doing excellent job. You are doing the excellent work, and you are, providing a lot to the society back. So, thanks a lot for giving your time to this conference.
Very much, Dr. Salahuddin and Dr. Ajit. I don't know because I could not hear you all properly. So I don't know how listeners have listened me and audience have seen my presentation. and because of this technical glitch. But still, I am sorry for that. And will hope that we can meet physically, and then we can have a good presentation.
Definitely, sir.
Yes, Dr. Said. Surely. We need to have again and again physical meet, and that's how we can. In fact, we should have a blended mode. Feature should be a blended mode activity rather than only a one-sided. So we can tackle both the things. In fact, this presentation was very nice. We can see the messages pouring in for your presentation. It was a great one. Thank you, Dr. Said. Thank you so much. Over to you, Dr. Raji.
Yes. So, sir, we will take a short break of 15 minutes and then we will start the poster presentations.
Yes, we can go ahead.
Okay? Yeah. Okay. So, see you in 15 minutes, everyone.
Thank you so much.
Thank you so much. Thank you.
Thank you very much. Thank you, brother.
Hello, good afternoon, everyone. My voice is audible. Hello.
Good afternoon, Dr. Nitesh. Yes,
Yes, sir. Good afternoon, sir. Good afternoon, sir. Thank you so much for a kind invitation, sir. How are you, sir?
Welcome, welcome. Thank you so much. So nice to see you. And we are happy that you have been part of our conference, national conference, and I'm sure your participation will add up more laurels and honors to this conference, Dr. Nitish.
Hi, Dr. Nitish. Good afternoon.
Hello, sir. Good afternoon. How are you?
I'm good, sir. How are you?
So really, it's a very great honor to me as I'm an evaluator in the international conference. So first of all, I would like to congratulate each and everyone, and a special thanks to Professor MD Dean sir for inviting me, for giving me honor to be part of this international conference. Thank you so much, sir. Thanks to all the members of the Al-Ameen College of Pharmacy, Bangalore. Thank you so much, sir.
Thank you, Nitish.
Thank you for accepting our invitation, sir. Thank you very much.
Welcome, sir. Thank you.
Dr. Ajit, Nitish is very young, dynamic, energetic, and if you see the progress of the Oriental University, a lot of the faculty of pharmacy is doing excellent, and the major contribution and the credit goes to Dr. Nitish.
I was just looking into his profile only, as Swati madam has said. He's doing really great, and he has done a lot of work. He's amazing works, amazing. Congratulations, and
thank you so much, sir, for kind words. But all the eminent personality like Salahuddin sir and you and we all are in a very same condition. So we are just promoting our profession, and definitely we'll work together in future. So,
definitely.
For the upliftment of the pharmacy profession, we will do whatever we can do. So thank you so much, sir, for kind words.
Thank you, Nitish. Are we going to start, Dr. Ajit?
Yes, we are going to start. I think madam is here, and
we are waiting. I'll just check with the other judge. Just a minute. I'll ask her to join.
Sure, madam. Sure. I hope all the participants, all the presenters are available here. Please raise your hand, guys. Chetan, I can see. Priya, I can see. Kindly raise your hands, guys, whoever is available. The presenters. We have selected 6 or 7 presentations for the conference. Okay, Vivek is also here. Yes.
Okay, Preeti madam is joining in just 2 minutes. She's having some problem with the Zoom, which is getting updated. She'll just join.
No worries.
Meanwhile, I'll do one thing. I'll just put up the list of, the order in which the presenters have to present, so that they are ready for the presentations.
Sure, sure, madam. Sure, madam.
So participants, in the chat box, I have pasted the order in which you will be presenting your papers. So please be ready. Priya Rajesh, you are going to be the first person to present. I hope your presentation is ready. You can please keep it ready on your computer. Yes.
Keep in the chat box.
Yes, I have put it in the chat box. Is it?
I think it's not for everyone, I think, ma'am.
Is it? Okay. I'll just put it to everyone. Wait, I'll put it again to everyone.
Yeah, Chetan is here. Yes, Dr. Anand Kishore is here. Okay, yeah.
Chetan has joined. Okay, he had requested us that he would be he might be a little delayed. Anyways, he can present in this order.
No problem.
So let me see if
Dr. Rajgopalan. Dr. Rajagopalan.
Yes, sir. Hello.
Are you here, sir?
Yes, sir. Yes, sir.
Great. Santosh. Mr. Santosh. Priya Rajesh, is she here?
Yes, sir.
Yeah, she's here. Okay. Sobarnika. Sobarnika, are you here? Tanmoji. Vivek is here. Chetan is here. Okay.
All right. I think both our judges are here. Shall we introduce them and start, or should
Yes,
we wait?
ma'am. Yes, ma'am. Yes. Priya, just wait for a minute. Let us introduce our judges. Then you can share the slides.
So good afternoon, participants and the paper presenters as well. So today we have with us two judges. One is Dr. Nitesh Jain. He's a very accomplished pharmacist and he's the Dean of Faculty of Pharmacy. He is Director, Drug and Disease Information Center, Oriental University, Indore. So he's a senior person with more than 14 years of academic experience. He has published more than 125 papers and he's got about 9 patents to his credit. So he's an accomplished researcher. So he'll be an ideal judge to judge your presentations. So to formally introduce him, he has won many prestigious awards like Eminent Professor Award, Inspirational Associate Professor Award, Visionary Principal Award, Inspirational Leadership Award, Young Scientist Award, Young Talent Award, Award for Excellency in Research, Outstanding Research in Pharmacy in Central India, for efforts made by him to bridging the gap between Indian and global standards. And he's also got the Academician of the Award Academician of the Year Award in 2018. So he is an approved PhD supervisor for Oriental University, Indore, and he has already awarded 4 PhD students. He has awarded the degree for 4 PhD students, and 3 have already submitted the thesis, and there are about 8 PhD scholars who are still pursuing their PhD under his guidance. So he has international exposure as plenary speaker in 22nd International Conference on Research in Life Sciences and Healthcare, which was held at Bangkok, Thailand. He has the following professional membership awards: membership of Society of Pharmaceutical Education and Research, membership of Indian Pharmacological Society, membership of Indian Pharmaceutical Association, membership of Indian Pharmaceutical Education and Sciences Lifetime, fellow membership of UPYA Foundation for Multidisciplinary Sciences, and he's also a reviewer or referee for a number of national and international journals: Natural Product Research from the Taylor and Francis Group, Journal of Advanced Pharmaceutical Research, JAPTR, American Journal of Cancer Prevention, American Journal of Pharmacological Sciences, Current Research in Biological and Pharmaceutical Research. He has done video recording on 5 expert lectures on pharmacology at Pharma World Studio, Educational Multimedia Research Center in Dr. H.S. Gore Central University, Sagar, under the planned project of NME ICT of MHRD Government of India. He is a reviewer in many other national and international journals. He's a member of LOC in DBT UGC sponsored projects, academia industry collaboration. Dr. Jain has organized many international and national conferences, World Congress, FDP, workshop, etc. as convener, organizing secretary and coordinator. He's also doing many social activities to promote rational use of medicine, and to provide unbiased information to all healthcare professionals and consumers by organizing seminars, drug information service activities, and adverse drug reaction monitoring awareness programs in community pharmacy, etc. So that's a very short introduction to Sir's achievements. I'm sure Sir is doing much more in his actual surroundings. And now to introduce, welcome, Sir.
Yes, thank you. Thank you for the introduction, brief introduction. Thank you so much.
Thank you, sir. So now to introduce another judge, we have with us Dr. Preeti Karwa, who is the head of the Department of Pharmaceutics at Al-Ameen College of Pharmacy. She has completed a B.Pharm, M.Pharm in Biopharmaceutics and PhD from Shivaji University, Government College of Pharmacy, Karad. She has to her credit 20 years of teaching experience in various subjects of pharmaceutics. Currently, she is working as a professor and head of the Department of Pharmaceutics in Al-Ameen College of Pharmacy, Bangalore. She has guided more than 25 PG students and is presently guiding 3 PhD students. She has published 13 research papers in various peer-reviewed international and national journals and presented more than 20 research papers in various reputed national and international conferences. She is subject expert at Rajiv Gandhi University of Health Sciences for postgraduate curriculum development program. And she is recognized as both UG and PG examiner in various universities. She is a lifetime member of professional bodies like IPA and APTI. So, Preeti ma'am, thank you so much for joining us at such a short notice. I think we will start. Yeah, we welcome you, ma'am, and I think we will start with our poster presentation program. I have already put up the order in the chat box for the students' references. So, Ms. Priya Rajesh, you can start with your presentation. You can start sharing your screen. About 10 minutes will be given to each of you, each of the presenters, of which in 7 minutes will be presentation and 3 minutes will be question answer session.
My slide is audible, sir. You are audible, but your voice is really low. Can you speak a little bit louder?
Now my voice is clear and please. Good afternoon. So now it's okay.
Your presentation is also not visible, Ms. Priya.
Now it's okay, sir.
No, it's not visible. You please reshare your presentation. Please share again.
If Miss Priya is having some difficulty, would you like to try for another 1 or 2 minutes, or should we move to the next person? You can present after that.
Okay, ma'am. I will present after that, ma'am.
You will present after that?
Yes, ma'am.
Okay. So, Mr. Rajgopalan, are you ready?
Yes, yes, madam. I am ready.
So, you please present.
Yes,
So,
yes.
Priya can present after you.
Hello. My PPT is visible. Hello.
Yes, it is visible and you are audible. Please go ahead.
Good afternoon, everybody. I am Rajagopalan. I am presenting my research topic on surface modification of 5-fluorouracil liposomal gel containing amide conjugate for management of actinic keratosis. So, what is actinic keratosis? It is the seed of cancer. It is the pre-cancerous of disease. It developed the skin cancers, that is particularly the serious type of skin cancer, squamous cell carcinoma. So this mainly affect the epidermal layer of the skin and the prolonged exposure to ultraviolet radiations. So 5-fluorouracil is a potent anti-cancer drugs. It's mainly chosen in combination therapy for treatment of numerous cancers, but it's having main disadvantages is the poor permeability in the skin. So with regard to improve the permeability, it was hypothesized in this study such an adjuvants with long hydrocarbon chain would give the desired hydrophobicity for optimal absorption of the drug molecule. So from the light of literature, a significant number of research work have been done on the topical drug delivery systems in the field of actinic keratosis, but there is a lacuna in the study to reflecting the synergistic effect. So in this study, modulated such a systems possesses a synergistic cytotoxic activity on lesion site and increase the localization accents and reduce the systemic toxicity. So for this purpose, the surface modified liposomal based 5-fluorouracil gel with the combination of gallic acid stearoylamine conjugate were used to achieve the intention. So first synthesize the gallic acid stearoylamine conjugate. The gallic acid stearoylamine conjugate was synthesized by using the Tris 2,2,2 trifluoroethyl borate reagents. The main advantages of this reactions here, direct amidation synthesis approach is used here, avoiding the tedious work of like column chromatography procedures by using the filtration purpose, purification purposes mainly used to call simple filtration protocol using the commercially available ion exchange resins for used for the purification purposes. So after the synthesized conjugates, it proved on the various characterization studies in NMR studies and mass spectroscopy studies, the gallic acid stearoylamine conjugate was prepared. So after proved through the various characterization studies, the cytotoxicity studies was conducted on the prepared 5-fluorouracils and also the prepared gallic acid stearoylamine conjugates. So it was showed that first individual 5-fluorouracil and the gallic acid stearoylamine conjugate cytotoxicity study was performed using A431 epidermal carcinoma cell line. It was observed that the 5-fluorouracil's IC30 value was 1 microgram per ml, and the prepared GASA conjugate was observed that 10 gram per ml. At the same time, the individual molecules also performed on the toxicity study using normal arcade cells. It was observed that the prepared GASA conjugates less toxic in arcade cells. So it is the perfect candidate for the anti-cancer drug delivery with reduced toxicity to non-target cells. So next, the cytotoxicity studies, combinations therapy of cytotoxicity studies was performed. The combination therapy of cytotoxicity studies in the combination of 5-fluorouracyls and prepared GASA conjugate was taken 9 ratio in the ratio of 5:1 to 1:5 based on the IC30 value of the individual 5-fluorouracil molecule and the GASA conjugate. So out of the 9 ratio, the cytotoxicity study was results that the 1:1 ratio was noted that increase the synergistic cytotoxic activity in A431 cancer cell line. At the same time, 9 ratio, the safety study was also observed in the normal arcade cells. The 1:1 ratio also observed that it's having the very less toxic in arcade cells. So finally, it was concluded that the results, the combination therapy of cytotoxicity study was observed twofold increase in the anti-cancer activity of 5-fluorouracils when combined with the GASA conjugate in the ratio of 1:1. At the same time, the low toxicity was noted in the same ratio, the 1:1 ratio. So in third phase, develop the formulations. So formulations developed, the surface modified liposomal gel was prepared using cost film iteration method. Using soya phosphatidyl choline, the soya lecithin was and cholesterol was used for this preparation of the liposome formulations. So using the 4 different ratio of lecithin and cholesterol was prepared. So out of this 4 ratio, the 7:3 ratio was observed that less vesicle size and good entrapment efficiency and zeta potentials and polydispersity index. So after the prepared liposomal formulations is converted to liposomal gel by incorporated the carbobol 934 gel base. So after preparation of liposomal gel formulations also characterized various studies, the spreadability, pH and drug content. It was also observed that the ratio of lecithin cholesterol ratio 7:3 was observed in the good spreadability, pH and drug contents. So a schematic diagram, it was showing that the prepared gallic acid stearoylamine conjugate, it was surfacely attached, anchored in the, because he is showing that the yellow color was the stearoylamine. So stearoylamine is the highly lipophilic in nature. It is interact between presence in the phospholipid molecules. So gallic acid is the hydrophilic regions. It is projected towards the hydrophilic domain of the liposomes. So this is the schematic diagram of the how can the gallic acid stearoylamine conjugate surfacely they attached, surfacely they anchored. So next study, the ex vivo skin permeation study and skin deposition study was performed using Swiss mice. So here the results was observed that the out of 4 formulations in compared with the marketer formulations, the LSGC formulations was observed that less flux value. It means maximum amount of drugs it is deposited in the skin and less amount of drug reaches the systemic circulations. But in marketer formulations, maximum amount of drug reaches the systemic circulations, very less amount its presence in the target sites. It was also, the graph is also shown that the skin deposition studies, the LSGC formulations maximum amount deposited in compared to marketer formulations only showing the less amount is deposited in the skin. So after that, it was the prepared liposomal gel was observed on the cytotoxicity studies. It is also that the IC30 value was observed that 0.1 microgram per ml. It is 50 times increase the cytotoxic activity compared to individual 5-fluorouracil drugs. After that, next study, the skin localization studies was performed on the confocal images studies using confocal laser scanning microscope studies. So the results was observed that the first group, it is used on the marker solutions, only 6-carboxyfluorescent marker solutions. The second group, the prepared 5-few liposomal gel formulations. So it was observed that the maximum amount of fluorescence intensity was present in the viable epidermal part of the skin, because the actinic keratosis, the origin area is the basal epidermal layer of the skin. So you can see the maximum intensity, it is present in the lower epidermal of the skin, the 25 micrometers, compared to the middle epidermis and also the upper epidermis. So it was confirmed that the results was confirmed that the maximum amount of drug is present in the target site, in the site of applications. Increase the localization of the skin. So in vivo, the distribution studies, animal studies, it was performed on the Institute of Nuclear Medicine and Allied Sciences in New Delhi. Using the gamma scintigraphy imaging studies and the distribution studies, the gamma scintigraphy images was observed that the maximum amount of activity of 5-fluorouracil gel formulations maximum extensively accumulate in the site of applications, the fluorouracil liposomal gel formulations compared to marketed formulations. So it was indicated the maximum amount of drugs in the presence, the target site of the application, and only it prevents the non-target locations in the body. So also the distribution study also confirmed that the prepared the liposomal gel formulation showing the higher skin blood ratio compared to marketed formulation. It was also proved that the maximum amount of drug reaches the site-specific delivery of actions, and only minimum amount of drugs present in the non-target locations in the body. So the gamma scintigraphy image and also distribution studies, it was proved that it was the prepared gel formulations, it having the good local action in the site of applications. So finally, the conclusions of this above work, we have designed and synthesized highly lipophilic molecule, the GASA conjugate, using the amidation approach. It was divided the tedious workups like chromatography procedures. So we utilized the scaffold for the development of 5-fluorouracil liposomal gel, which exhibited enhanced biocompatible potential, also a safer approach for the treatment of actinic keratosis comparisons to marketed one. So we anticipated that the future works is useful for the ex vivo permeation study using human cadaver skin, and also using the in vivo distribution study using the xenograft model, the immune suppressed models, completed, sir, myself. Hello.
Yes, Mr. Rajgopal.
Yes, ma'am.
So you have questions for him, sir?
Yes, please. Okay. So Raju Gopal, really you have done a great innovation, and you have covered almost all the aspects of research. Especially you have done in vivo, ex vivo, animals study. You have prepared some conjugation. So overall research was really very innovative and very like what we can say, you have done something different. So I have some questions from you. What is the correlation between IC50 value and cytotoxicity study?
So IC50 value is how much the cancer cells are killed in the 30% reduction. This is the IC30 value, sir.
Okay. And another question is, almost there are many researchers who are working on the cancer. They are finding difficult still cancer is not curable, right? Because of metabolic pathway of cancer cells and normal cells are almost same. So
Yes,
as you have
sir.
some liposome gels, right? So what is the new findings as if we compare with the traditional or the conventional approach, whatever we are using for the treatment of cancer?
Sir, in a conventional approach, marketed already available existing the 5-fluorouracil cream, the brand name Flonida cream in worldwide already existing, but it is available in cream formulations. The main disadvantages of these formulations, the 5-fluorouracil is having the poor permeability in the skin. So the marketed available the cream formulations, very less amount of drug only reaches the target sites. This is the major disadvantages of the 5-fluorouracil cream formulations. But in our novel drug delivery system like liposome formulations, it is very very good advantages compared to conventional, because it having the controlled drug delivery action also, given the sustained action also, and also same times it given the increase the potency also, and also it give the good local actions, increase the local action, and also its main advantage, another one, it should be biocompatible with our human body lipids.
Okay. So, dear, just tell me one thing, that what will be the penetration power of that particular liposomal gel? As we talk about the nanomedicines, nanoparticles, so what is the correlation between absorbance versus cytotoxicity?
Yeah, because your main part is your main role is the prepared the gallic acid steer element conjugates. It's having the highly lipophilic molecules. It having the long hydrocarbon chain. Because these long hydrocarbon chains, it give the increase the permeability of the systems. So after the administer the system, the surface attached the gallic acid steer element conjugates, it reaches the lipids. Because it is contained the highly lipophilic nature, it is easily bind with our intracellular lipids. It produces the intracutaneous depot effects. So this intracutaneous depot effect, it is easily reaches the target sites and give the increase the local actions. So it increase the permeability of the systems to avoid the permeability of the 5-fluorouracil drug also, and reaches the target sites, increase the local action.
Your work is so vast. There are many questions, and really you have given a lot of time, so it's very difficult to judge within a 1 minute or 2 minutes.
Yeah, exactly.
But I really congratulate you for doing a very innovative work. So really, you deserve compliments for this. Keep continue. Thank you. Ma'am, over to you.
Yeah.
Please.
Mr. Rajgopal.
Yes, madam.
Yeah. What was the particle size you got for this liposome, whatever you have prepared?
Liposome, 1 minute, ma'am. Particle size.
Yeah.
Because
that liposome you have prepared and then incorporated in gel.
Vesicles, vesicle size 158.4 nanometer. Yeah.
Okay. 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 So you have given that vesicle size and structure also you have given. How you have seen that?
What, madam?
Structure. That exactly liposomal structure you have got. SEM studies or TEM studies?
I am done at TEM studies, transmission electron microscopy study was performed, but I am not showing you that.
Yeah, that's what I have seen.
Yeah.
Okay. Okay.
Okay. Thank you so much,
Yes.
Mr. Rajgopalan. Can we move ahead?
Yes.
Okay. So, Miss Priya, are you ready with your presentation now? Miss Priya?
Yes, ma'am.
Are you ready with your presentation now?
Yes, ma'am.
So you can present now. Just one small request. The presenters can keep their camera on and preferably do not exceed the time limit.
Yes.
All right? Yeah, because then we'll get delayed. Yeah, you may please start with the presentation.
Audible, ma'am?
Yes, it is audible, but the voice is a little low.
Good afternoon to everyone. Today my topic is principal and
Okay. Can you make it full screen, Miss Priya?
Screen share can go.
No, it is not full screen right now. So you can just reshare the full screen.
Now it's okay, sir.
No, you go to full screen option, my dear. Present from the beginning. That
PPT.
I think then you have
Slide
to
share, PPT presentation.
please reset the presentation full screen. Reset the full screen.
Try to press Shift and F5 on your laptop or computer. Shift and F5 on your keyboard. Shift key and F5 on your keyboard.
Now it's okay, ma'am.
No, my dear, we can't see it as a full screen. It is still, go to slideshow if you know how to go to slideshow.
Ma'am, it's visible if she may continue with this.
Yeah, all
Yeah,
right.
she can continue. Yeah, yeah. You can continue, Miss Priya, with this. No problem.
Everyone, today our present topic is principle and applications of proteomic science.
Yeah, boy,
Your voice is not audible.
Now it's okay. Hello, good afternoon. Okay, ma'am.
Yes.
Yes, please continue.
Okay, ma'am. Thank you for the chance. What is a proteomics means? It is a nothing but proteomics. It's a large dimension study of the protein. So proteomics it's a nothing but it is a large dimension study of proteins. Proteins are essential for the parts of the living organism. It needed for the more function. For example, we make the structural fibers and muscles to enzyme catalyze the digestion of the food, synthesizing and replicating the DNA. The term proteosome was coined by Australian PhD students Mark Williams in 1994 in Italy. So next we want to study in the DNA. It is called genomics. The conversion of the DNA to RNA, it's called a transcription process. To study of the RNA, it's called a transcriptomics, and the protein means study of the proteomics. What is the DNA sequencing? The DNA sequencing is the process of the identifying the nucleic acid sequences in order of the nucleotide in the DNA. It contains mainly 4 bases: adenine, guanine, cytosine, and thymine. Always adenine is paired with the thymine and guanine is paired with the cytosine. Next step involved in the DNA sequencing: sample preparation, PCR amplification, amplicons purification, sequencing of pre-preparation, and DNA sequencing, and finally we want to analyze the data. DNA sequencing in the identification of genetic risks. One new sequencing technology involved in the watching the DNA polymerase molecule as the copy of DNA, the same molecule that make the new copies of DNA in our cells with a very fast moving camera and microscope, and incorporating in the different color bright dyes, one of each letter: adenine, thymine, cytosine, and guanine. This method is provide different and very valuable information. And another new technology in development entails use of the nanopore sequence in the DNA. And another one technology of the identifications of the genetic disease, it is the used in the nanopore based technology in the DNA sequencing involved in the threading single DNA strand throughout extremely tiny pores in the membrane. DNA bases are read at a time they squeeze their nanopore. The bases are identified by measuring the difference in their effect ions and the electrical current flowing in the pores. And next is the metabolomics in the identification and the management of the pre-diabetics and the diabetics. What is the objective of this topic? Means to conduct the systematic review, cross-sectional and prospective human studies evaluating metabolite makers identify using high throughput metabolomics sequencing on the pre-diabetes and the diabetes. What is the research design and the method? I determined from the Medline and the MPACE database throughout August 2015 year. It was conducted in the qualitative review, cross-sectional and prospective studies. Additionally, meta-analysis of metabolites marker with the data estimate from the last 3-year prospective studies and type 2 diabetes risk were conducted, and a multivariable adjusted relative risk of type 2 diabetes were calculated. Study specific ST difference in the different given metabolite. What is the result means? Identify the 27 cross-sectional and 19 prospective publication reporting association of the metabolites and pre-diabetic or type 2 diabetic, carbohydrate, lipid and amino acid and aromatic amino acid metabolites were higher in the individual type 2 diabetes compared with the control subject. What is the conclusion means? In this study, high throughput metabolomic, several blood amino acid appear consistently associated with risk of developing diabetes. Next topic, genetic mapping in, what is the genetic mapping in the type 1 diabetics? HLA is nothing but human leukocyte antigen. The risk of the developing type 1 diabetes is increased in the certain variants of the HLA-DQA, HLA-DQB, and HLA-DRB1 genes. So it is a genetic manipulation in it is acquired in the HLA gene. The gene provides the instruction to making the protein that play in the critical role in the immune system. The HLA complex help in the immune system to distinguish in the body own proteins from the proteins made by foreign invaders such as the virus and the bacteria. Type 1 diabetes is generally considered to be the autoimmune disorder. The autoimmune disorder occurs when the immune system attacks the body own tissues and organs. For unknown reason, in the patient with the type 1 diabetes, immune system damage the insulin-producing beta cell in the pancreas. Damage to this cell impairs with the insulin production and lead to the sign and the symptoms of the type 1 diabetics. HLA, human leukocyte antigen gene is included DQA, DQB, and DRB1 type of the many variation in the individual certain combination of this variation is called haplotype. I given in the imagine what is the type of the haplotype: type 1, type 2, type 3, and type 4. In the we want to find the image differentiation. Next, role of the genome sequencing and the identification of emerging SARS-CoV-2. Despite worldwide vaccination, COVID-19 pandemic continues SARS and numerous variants. Since first identification of the novel variants in the Omicron on November 24th, 2021, it is immune compromised patients South Africa. The variant has Delta predominant linkage in the South Africa and has quickly spread over the 14 countries. He provided the initial molecular characterization of Omicron variant through analyzing large number of mutations, especially spike protein receptor binding domain. When there is the potential effect of the viral infectivity and reduced immunity, our analysis indicates the Omicron variants have subclade may envelope the clade 20b instead of currently dominant Delta variant. In addition, we have also identified the mutation that may affect the AC2 receptor and or its AC2 receptor antibody binding. Tools for metabolite profiling and early detection of the disease. The emerging field metabolite, large study of small molecular metabolite from the body fluid and tissues are detected quantitatively in the single steps. Promises the impairs potential early diagnosis, therapy monitoring, and for understanding these pathogens of many disease. Metabolomics methods are mostly focused in the information-rich analytical technique for the nuclear magnetic resonance spectroscopy and mass spectroscopy. Analysis of the data from the high-resolution method using advanced chemometric approaches provided the powerful platform for the translational and clinical clinical research and diagnostic application. Next, and finally, we want to see the applications of the proteomic science. What is the application means? We want to use using in the proteosome mining, protein expression profiling, post-translational modification, protein-protein interaction, and the structural proteomics and the functional proteomics. In the proteosome mining, we want to use in the drug discovery, target identification, and the target validation, differential display. In the protein expression pooling, we are using the medical microbiology, signal transduction, disease mechanism, and post-translation modification. We are using the glycosylation, phosphorylation, proteolysis, protein-protein interaction, yeast to hybrid, co-precipitation, phase display, and structural proteomics, organocomposition, subproteosome isolation, and protein complexes, and finally functional proteomics, yeast genomics, affinity purified protein complex, and mouse knockout method. These are the applications of the proteomics. Thank you for the.
Okay. So Priya, you have given a wonderful introduction about proteomic science. So just I want to know about what will be the first step if we are going for drug discovery, if we are using that proteomic science in drug discovery. And one compliment for your voice. We are just thinking that we are listening about the news of pharma. So your voice is so, soothing, and definitely it was a very good explanation. But I just want to know if we are going for drug discovery of new compounds, as you said that we are using in COVID-19 pandemic regarding vaccination preparation, right?
Yes,
so what was the first step, especially if we are using that particular science in drug discovery? No idea?
No, sir.
Okay. Over to Preeti ma'am.
Yes, Priya.
Yes, ma'am.
Is it possible to analyze hydrophobicity of larger set of protein IDs in proteomics?
Proteomics?
Is it possible to analyze hydrophobicity of large set of protein IDs or sequences?
Only we are using the small molecules only we are using them. It's large molecule, it's not possible.
It is not possible?
Yes,
okay, small molecule, whether we can recognize or whether we can know hydrophobicity of that molecule. Okay, you don't know, I think.
Yes, ma'am, I don't know.
Have you heard word transcriptomics?
Yes, ma'am, transcriptomics. It's a study of RNA.
So, which is easy? Transcriptomics is easy or proteomics is?
It's proteomics is easy, means proteins are large molecule. It's a study, it's in the enzymatic reaction. So it's proteomics, it's a study of easier than our transcriptomics.
Okay.
Okay, I think, thank you, Priya.
Yeah, thank you, Priya.
Thank you, ma'am. Thank you, ma'am. Thank you, sir.
Thank you, Priya. I think next presenter is Mr. Santosh. Are you ready, Mr. Santosh? Santosh, are you there today? I don't think Santosh is present today because I think Ajit sir, you were also asking for him. He did not reply, I suppose.
No, he did not reply, madam.
So then shall we move on to the next person?
We can go to the next.
Yeah, Shaubarnika. Ms. Shaubarnika is? Are you there? Shaubarnika is? Are you there? No, I don't think she's also there.
Tanmoji?
Tanmoji?
Chetan?
Yes, sir.
Chetan, please go ahead. Chetan, please go ahead with the presentation.
समझते हैं ना, तुम इंटरनल तो कराओगे, कॉपी का देख लेंगे, मार्क्स चढ़वा लेंगे मार्क्स।
Sir, my slides are visible, sir?
Yes.
Visible. Make it full screen and go ahead.
Okay, thank you.
And Vivek, be ready.
Good morning, good afternoon, sir, everyone. I am myself Chetan Kumar. I am presenting a review article on role of herbal remedies in reduction of symptoms. Sorry. In the reduction of symptoms in the patient diagnosed with the anxiety disorder. First, I am going to introduce anxiety is the psychological mental state which is sorry, sir.
Please go ahead. Please go ahead, Chetan.
Okay. Anxiety is psychological mental state of expectation of future upcoming negative events, yet preparation for future imaginary threat, and the patient suffers from symptoms such as vague fear, worrying, disproportionate level of stress, awareness, muscle tension, and physical symptoms like excessive sweating, twitching, and irritability, tension, restlessness, palpitation, and poor sleep quality, and it impacts well-being and health status of patient. Based on psychological feature of anxiety, it was classified in three types: threat, state, and situation-specific anxiety. As a diagnostic and statistical manual of mental disorder, as per fourth edition, and International Classification of Diseases, ICD-10, anxiety is classified into 7 types: social anxiety disorder, generalized anxiety disorder, agoraphobia without panic disorder, acute stress disorder, obsessive compulsive disorder, and post-traumatic stress disorder. And etiology of anxiety, as per the early studies, anxiety is mainly due to the impairment in the neurotransmitters such as epinephrine, serotonin, dopamine, and GABA, and also anxiety associated with the increased oxidative stress and depletion of vitamin E. Then also it is causes due to the environmental factors like personal relationship, job and school, and genetic factors like family history, inherited genes causes the anxiety. Then in recent survey, it is clinical, and it is suggests that anxiety also associated with medical conditions such as hemodialysis, COVID-19 pandemic, and acute coronary syndromes, advanced cancer, diabetic mellitus, paired nerve injury, cirrhosis, chronic pain, and inflammatory bowel syndrome, etc. The pharmacological targets of this monoamine system, GABA system, glutamate system, and endocannabinoid systems and neuropeptide systems are the pharmacological target for the anxiety. And treatment, there are three different types of treatment are there. There are pharmacotherapy, psychotherapy, and phytotherapy. Pharmacotherapy is the treatment with the medicine. Psychotherapy is considered as an effective therapy of the psychiatric disorders such as anxiety and depression. And phytotherapy is treatment with the various herbal drugs. The clinically approved recent herbal drugs, although various effective drugs and herbal drugs are available for to reduce the anxiety, because their various side effect and adverse effect, many herbal drugs are discovered in recent years with the aim to provide high effective anxiety herbal drugs with the minimum side effect. Various preclinical and clinical research were carried out in recent year and discovered various herbal drugs. Among many clinical approved herbal drugs, some drugs such as valerian root, lemongrass, ashwagandha, passion flower, and cannabidiol, and L-theanine, and citrus aroma, nanocurcumin, cannabis, and lavenders are discussed in below. The first one is valerian root, which is scientific name is Valeriana officinalia and family Cyprofoliaceae. It contains chemical constituents like volatile oil, ketones, phenols, and esters such as valproates, arginine, and its root contain essential oil like valerianic acid. And its mechanism of action of valerian root is it binds to the GABA receptor, acts as a GABA agonist, and improves the normal sleep time. And it is used for sedative, sleep disorder, anxiety, stress, muscle relaxant, and other conditions like pain, inflammation. arthritis. It is approved in 2020. And next one is curcumin. The general name is curcumin. And scientific name is Curcuma longa. It is belongs to the family Gingiberaceae, and it contains the chemical constituent curcuminoid, curcumin, and dimethoxycurcumin, etc. The mechanism of action of curcumin is act through a serotonin and dopamine receptor. And medical uses of curcumin are prevent from many CNS disorders like inflammation, oxidative stress, psychological stress, anxiety, depression, and neurodegenerative disorder. And it is approved in the 2019. And Brahmi is generally known as Brahmi, and scientific name is Bacopa manari, and it belongs to the family Scopholaceae. It contains chemical constituent brahmini, herpestine, alkaloid sulfonates, bacoside A and bacoside and glycosides. Mechanism of action of it is bind to a GABA receptor. It has a GABA agonist activity, and its medicinal activity uses is antioxidative, anticonvulsant, antidepressant, anti-epileptic, memory enhancer, sedative, and anti-anxiety agent. It is clinically approved in 2021. And L-theanine.
Yeah, you may discuss in short. I think there is no need to write everything about it. So if you can correlate these things with the anti-anxiety, that will be better, right?
Yes, sir.
There is no need to give much more explanation. You can give a name of the drugs and what is the basic features. That's why we are using as anti-anxiety.
Yes, sir.
Don't read everything like chemical constituent, medical uses and all, right?
Yes.
You can give overview about this particular review of literature or review whatever you have done on herbal drugs for anxiety, right?
Yes.
Okay, continue, please.
Next one is ashwagandha. It has chemical contents. Sorry, sir, I just prepared like that only. Can I continue with that one?
But you can cut short.
Move on. Yes.
You can just explain its medicinal.
Yes.
Or in anxiety, how it is useful.
Yes.
Because
you need
that
not
is
give
what is
everything.
required.
Yes. Your title says.
Okay, come to conclusions. Okay.
Sir, just I prepare slide with only drug name and scientific name. That's all.
No, I agree. You have given a good presentation and you have a brief story, but as we have limited time for a discussion, that's why I'm saying that there's no need to read everything, right?
No. Just I prepared like this much only, sir. And next it is the end.
Okay, great. So Santosh, right?
All right. So if you have any questions, you may ask him.
He is Chetan, sir.
No, Chetan.
Sorry, Chetan Kumar. So Chetan, just tell me about one thing. If we are going for herbal medicine, right?
Yes,
there are very misconception in the society that herbals are always safe. What about your opinion about the herbals medicine for the treatment of any disorder?
It is better than allopathy medicine. It is have more effects, sir.
This
as
is
compared to
common comments that herbals are always safe, better than the allopathic medicine. How we can say there are some emergency condition in which only we can use allopathic medicines, right? We cannot recover with the Ayurvedic. If we go for the herbal medicines, now patients will die, right?
Yes, it is a less side effect they have, sir. Herbal medicine.
Side effect we can say because this is a very slow process and herbals are comparatively safe. But what about the misconception? Herbals are always safe or the entirely safe?
No, it is not always safe, sir. Sometimes it also has dependent on dosage how it will take.
Just tell me only last answer. If any compound having anti-anxiety activity, what basic phytoconstituents will be responsible for anti-anxiety activity as per your review of literature or review?
It has flavonoids. If it has antioxidant property means.
Okay.
Anxiety due to the oxidative stress, it can be cured.
Okay. Okay. Thank you. Over to Preeti ma'am.
Thank you,
Yes,
sir.
Chetan.
Yes,
Just one question to you. Can anxiety be treated without medication, even herbal and even allopathic?
yes, ma'am, I can't hear.
Can anxiety be treated without medication?
Yes, ma'am,
But
it is
no herbal.
psychological treatment, ma'am. Exercise, music therapy is there, and meditation is there. You can.
That is what it is best treatment for anxiety. That is cognitive behavioral
Yes.
Agree.
therapy.
Yes. Physical exercise are the best treatment. It is considered now. It is.
Okay, Chetan, next time.
Okay. Okay. Thank you.
Thank you, ma'am.
Yeah. All right, Mr. Vivek, are you there? Mr. Vivek, are you there? Yes. So you may please present.
Okay. And the slides are visible.
Make it full screen, dear.
No, sir.
Make it full screen.
Yes, sir. I have made.
Go on slideshow.
Please make it full screen, Vivek.
Yes, sir. I have done.
Okay,
Now?
no problem. You may continue with this. We are able to see.
Or reset the full screen. I think you have to reset the full screen, Vivek. Please do that.
No, ma'am.
No problem, Vivek. You continue with this. No problem. That's fine. Yeah, now it is visible.
Okay. Good afternoon, everyone. I'm second year. I'm from Pharmacy College of Pharmacy. Today, I am going to present my review article on current perspective in the management of depression using off-label drugs and advanced drugs. These are the contents I am going to explain right now. Introduction, diagnosis, off-label drugs, and approved drugs for depression. Introduction. Depression is a mood illness that occurs on a regular basis.
Dear, no need to define the definition of depression. Come to the topic or maybe discussion which is related to your main title.
Yes.
Okay, sir. This is the etiology. Nowadays, it is most common in most common disorder. Hello.
Yes, go ahead. Go ahead.
Okay, sir. Nowadays, it is a most common disorder. Alopecia also causes depression because of loss of hairs, and nowadays COVID-19.
Participants, please put your mics on mute, except Mr. Vivek, except the presenter. Other.
Nowadays, COVID-19 in COPD patients is also some COPD patients will be more subject to mental diseases like symptoms and disorders such as traumatic stress illness and fear, anxious, like that. Abuse as age becomes older, and physical, sexual, and mental abuse might increase susceptibility to depression. Depressive disorders are much more common among the elderly. Other factors such as living alone and lack of social supports might aggressive
Your
problems.
slides are not moving, so I think you may move your slides with your explanation.
Yes, sir. I am moving the slides.
It's not moving. It's not moving, Vivek.
I think you exit your full screen and you present it like this only. Yes.
Yes, ma'am. Death, sadness, or grief caused by the death or loss of a loved one may raise the risk of depression. Nowadays, females are twice as likely to men and to suffer from depression. Nobody is sure why hormonal fluctuations that women experience at different phases of their lives may be an influence. Genes. There may be a familial history of depression. Depression is a complex characteristic suggesting that there are likely many different genes, each having a tiny effect rather than a single gene that influences illness risk. The genetics of depression, like those of other mental disorders, are more intricate than those of simply inherited illness such as Huntington, chorea, or cystic fibrosis. In cancer, in individuals with common malignancies, major depression is likely to have worse chance of survival and also linked with hair loss and pain or for chemotherapy. Next is diagnosis. Two order frameworks are broadly utilized. The fifth version of Diagnostic and Statistical Manual of Mental Disorder, DSM-5, created by the Association of American Psychiatric and Worldwide International Classification of Diseases and Related Health Problems, 11th correction, created and as of late refreshed by WHO. This is a new method of diagnosis using electroencephalography readings. The non-stationary and non-linear functioning of brain signals is depicted by EEG recordings. There hasn't been much progress more on automated identification of depression using electroencephalography signals. In comparison to a healthy participant, depressed patients as compared to not depressed individuals, depressed patients had more activated signals. This is the normal signal of left hemisphere and normal signal of right hemisphere, non-depressed people signals. This is the depressed people signals of left hemisphere and right hemisphere. We can see the fluctuations that as compared to normal depressed peoples. Other methods for diagnosis are formalized assessment and intensity evaluation, psychiatric diagnosis, and physical diagnosis. These are the off-label drugs which are used for the depression. Duloxetine and milnacipran, which are normally used for chronic pain of fibromyalgia. Mechanism of action is increases the dopamine level. As a result, it causes selective serotonin and norepinephrine reuptake inhibitor. Bupropion, it is normally used for cessation of smoking. Norepinephrine and dopamine reuptake are slightly inhibited. Dopamine reuptake inhibition is said to be quite strong. Venlafaxine, this is normally used for complex pain syndrome, for migraine, and for diabetic neuropathy and fibromyalgia. Mechanism of action is powerful serotonin reuptake inhibitor. Paroxetine, dysphoric, it is used for dysphoric premenstrual disorder, night sweat associated with menopause, hot flashes, and mechanism of action is it inhibits the serotonin reuptake transporter. These are the approved drugs for depression. Esketamine nasal spray, immediate release on March 5th, 2019. The US FDA recently accepted Spravato, which is a nasal spray for a treatment of depression in individuals who have tried but failed to benefit via existing antidepressant medication. Individuals with major depressive disorder who have failed to respond to treatment despite at least 2 attempts at appropriate doses of an antidepressant for an appropriate duration in the current episodes are considered treatment-resistant depression. Mechanism of action, the mechanism where the esketamine exerts its antidepressant effect is unknown. Normally, it is the antagonist of N-methyl D-aspartate receptor. Side effects, the most prevalent adverse effects seen by individuals treated with Spravato in clinical studies are dissociation, dizziness, nausea, sleepiness, vertigo, reduced sensation, anxiety, and raised blood pressure, vomiting, and feeling drunk. Brexanolone IV injection, it is immediate release on 19th March 2019. The United States FDA, US FDA has authorized Zulresso injection for intravenous use in adult women suffering from postpartum depression. This is the FDA's first medicine authorized for postpartum depression. The mechanism of action of Brexan alone is not fully known. Side effects in clinical tests, drowsiness, dry mouth, unconsciousness were among the most common adverse effects reported. Medical providers should consider altering a patient's medication plan, including stopping Zulresso if their postpartum depression worsens or they exhibit sudden suicidal thoughts and acts. These are some of the references I refer. Thank you so much.
Okay, dear Vivek, you have given a lot of explanation about the off-label drugs and new drugs. So this was something new. So I think it's okay. Thank you for the information.
Thank you, sir.
Mr. Vivek,
Yes, ma'am.
is off-label use of drug is common?
Yes, ma'am. Not really common. It can also be used by
But then by
trial and error
government
methods.
approval is not obtained to convert off-label use to
No, ma'am.
on-label uses.
As per the mechanism of actions, it can be used, ma'am.
Yeah, it can be used, but is it very common?
Yes, ma'am.
Can physicians use? Is it their legal practice? They can use this off-label drugs?
I don't know exactly, ma'am.
Okay.
Thank you, Vivek. Next.
Thank you.
I think that was the last presentation. Our other presenters have kind of backed out. They are not really present in the meeting. We've been trying to contact them. So I think this was the last presenter. So
Once again, I would like to thank you, Al-Ameen College of Pharmacy, Bangalore, especially Professor M.D. Salahuddin sir, Dr. Ajit Singh sir, and Swati Mittal madam, and my co-evaluator, my colleague Preeti madam. Thank you so much for inviting.
Thank you, sir. Thank you.
Thank you, sir. Thank you so much. It was very nice to have you on board, and thank you, everyone. I would like to thank our presenters also. Dr. Rajgopalan, Priya, Chetan Kumar, and Vivek. You put your good efforts, and of course, I think three presenters have backed out, but that was fine. I think it was really cool and really good. All was amazing, and the entire conference is I think I should appraise our audiences. Even in the poster presentation in the lunchtime, we have a very, very good number at this time during the examinations. If everyone is preparing for the exams and they are preparing for the practical exams and they are busy in their academics, and still we have a good number. And since morning we have top full 100 and plus in the YouTube channel, and it was amazing, successful conference, I would say. So thank you, everyone. Thank you, Professor Salahuddin sir, for the opportunity and the collaboration, and thank you, Swati madam. Thank you, Dr. Nitesh, and our other speakers, and thank you, Professor Preeti madam. So
All credits to the young dynamic team of Al-Ameen College of Pharmacy, and we wish you a grand success of the international conference. So, sir, we'll keep in touch and we'll work together for upliftment of the pharmacy.
Definitely, sir. Definitely.
Thank you, Salahuddin sir. Thank you so much.
Thank you.
Thank you so much.
Thank you, everyone. And yes, sir.
Conclude, and it was a great day, and I must say that another initiation in the field of research and the knowledge sharing that we have done.
Yes.
So we keep seeing you doing an excellent work. I'm sure you keep spreading. Thank you so much.
Thank you. And one more very successful conference we have conducted. It is amazing, So I will hand over to Professor Swati madam to conclude the conference and give a vote of thanks.
Yes.
Good evening, everyone. It has been a great pleasure having all of you over here. So though we have already exchanged so many thank yous and gratitude to everyone, I would like to express my formal gratitude to everyone. So on behalf of Al-Ameen College of Pharmacy and CliMed Research Solutions, I extend my heartfelt gratitude to our esteemed speakers, Dr. Manoj Kadekar, Professor, School of Medical and Allied Sciences, KR Mangalam University, Gurgaon, Dr. Preeti Sangwe, Associate Professor, NMIMS University, Shirpur Campus, and Dr. Saeed Ahmed, Professor, Jamia Hamdard University, Delhi. Thank you, speakers, for making time from your busy schedules to deliver such informative lectures and making us wiser on principles and applications of proteomic sciences. My sincere thanks to Dr. Ajit Singh, CEO and co-founder of CliMed Research Solutions, and Dr. Mohammad Salahuddin, principal of Al-Ameen College of Pharmacy, who gave us the opportunity to organize this online conference on a very futuristic topic. I thank my colleagues for cooperating with me at all times for successful organization of the conference, Ms. Manjula, Ms. Deepa, Ms. Kavana, Ms. Bindya, as well as Mr. Dharmendra. I extend my heartfelt thanks to the judges who have spent their valuable time evaluating the scientific abstracts. Also, there should be a wide round of applause for all the participants who have made the seminar possible. I hope I have not missed out anybody in my compilation of the people to whom I would express my gratitude. Yes, I did miss out Dr. Muthukumar. Muthukumar has been there with me all the time throughout, right from the beginning. I'm sorry, sir, I missed out your name. I mentioned all the others except sir. Yeah, so I think we come to an end of our wonderful session. It has been really very informative for most of us. And the speakers were very, very knowledgeable people who have shared immense information with us. Thank you so much. Thank you, Ajit Singh, sir, and thank you, Salahuddin.
Thank you, madam. Thank you, everyone. Thank you, sir. And if everyone is available, we can take a screenshot. If everyone can open their, video cameras. Yes. I request everyone to put on their cameras, if it is possible.
All participants?
Yes, all the participants.
Yes, all your smart faces should be on screen.
Yes.
Absolutely.
Yes, please, everyone.
Confident people will be on screen.
Yeah, true. Okay, so thank you, everyone, and I hope to see you soon, very soon, in our upcoming events and other conferences, workshops, and courses. So, and looking forward for further collaborations with all of you. Thank you very much, and take care. Have a good day, and bye bye. Thank you, sir. Take care, sir. Bye bye.
Take care, everyone. Bye bye.
Thank you. Thank you.